Yeah. ঠিক করে দেন না হ্যালো চন্দন
হ্যাঁ স্যার
চন্দন
হ্যাঁ স্যার শুনতে পাচ্ছি স্যার।
Hello and good evening to everyone. I am Rajina Sultana.
চন্দন স্ক্রিনটা ইউ এন্টার দ্য স্ক্রিনে ঢুকতে হবে না
হ্যাঁ স্যার
আচ্ছা এন্টার দ্য স্ক্রিনে ঢোকার পর আচ্ছা শেয়ার নাও আই গট ইট শেয়ার
হ্যাঁ স্যার।
আমি কি এখন স্ক্রিনটা অন করে দেব স্টপ প্রেজেন্টিং
না স্যার একটু পরে
দাঁড়াও স্টপ প্রেজেন্টিং দেখাচ্ছি কেন প্রেজেন্টিং ঠিক আছে আমাকে হ্যাঁ আমি টার্ন অফ করে রাখছি আমাকে একটু জানিও
হ্যাঁ স্যার হ্যাঁ স্যার
Okay, Jaydeep, I am.
Hello and good evening to everyone. I am Rajina Sultana, Assistant Professor of Zoology, Kalna College. Welcome you all on the very first day of 2-day state-level webinar named COVID-19 Molecular Aspect and its Possible Magnitude, jointly organized by Department of Zoology and Department of Botany of Kalna College with collaboration with IQAC, Kalna College, Kalna, Purbo Bardhaman, West Bengal. I humbly request all the participants to please put your audio and video off during the entire session and do not share your screen. I would like to welcome our innovator, Professor Debashish Bhattacharya, our Teachers Council Secretary, Professor Dipankar Roy, our webinar convener, advisory committee, organizing member, and all the faculty and the participant of various college and university institutes. welcome you all on the very first day of 2-day state level webinar. Now I'd like to invite one of our convener, Dr. Joydeep Acharya, to say some few words about today's session. Over to you.
Good evening. Myself, Joydeep Acharya. I am taking the opportunity to introduce this webinar highlighting today's activity. Hello, am I audible to all?
Yes.
Yes. Yes.
Today's date, being 22 Shravan, holds special significance for every Bengali. Tagore, one of the greatest visionaries from Bengal, has transcended the borders of science and literature to create a unique regime of his own. His song, Akash Bhora Surjo Tara, Vishwa Bhora Pran, speaks of his immense curiosity about astronomy and life science. So I begin this webinar paying homage to the bird of Bengal on his 79th death anniversary. This webinar, entitled COVID-19 Effects and Possible Managements, has been initiated jointly by the Department of Zoology and Botany of Kalna College in collaboration with the IQSE under the patronage of Honorable Principal Sir Dr. Tapus Samanto in context to the present global crisis created by the simplest creature, which is the RNA virus. The lectures of this webinar promises to equip us with encouragement to combat the challenges imposed by this pandemic. I am glad to say that there are 415 participants in this webinar. 196 are UG students of different colleges, 69 are PG students, and 122 faculties of different institutions of this state. I heartily welcome all of you in this 2-day state level webinar. কারীদের আমি আমাদের এই দুদিনের ওয়েবিনারে আন্তরিকভাবে স্বাগতম জানাই। Webinar constitutes of 2 lectures each day, and after completion of each lecture there will be a session. The interested participants are requested to post their questions in their respective chat box. Questions would be passed on us. I repeat, please do not ask any question, just type it in your chat box. We will pass the relevant question to the speaker. Now, abiding by the schedule of our webinar, the first lecture entitled COVID-19, the molecular information, will be delivered by Dr. Sudhit Shekhar Mukhopadhyay, Department of Biotechnology of NIT, Durgapur. It will start on 5:50 p.m. from 6:30, and it will be presented by a medical professional who is a consultant physician associated with Medica Super Speciality Hospital, Kolkata. Lecture is COVID-19 Diagnosis and Prevention. স্টুডেন্টদের জন্য একবার বাংলায় বলে দিই, প্রথম বক্তৃতার বিষয় হলো কোভিড-19 দ্য মলিকুলার ইনফরমেশন, অর্থাৎ কোভিড-19 সংক্রান্ত আণবিক তথ্য। এটি এনআইটি দুর্গাপুরের বায়োটেকনোলজি বিভাগের প্রফেসর ডক্টর সুদীপ শেখর মুখোপাধ্যায় মহাশয় সহজ সাবলীল ভাষায় আমাদের বুঝিয়ে বলবেন। 5:50 শুরু হবে। দ্বিতীয় বক্তৃতার বিষয় হলো কোভিড-19 ডায়াগনোসিস অ্যান্ড প্রিভেনশন, অর্থাৎ কোভিড-19 রোগ শনাক্তকরণ এবং প্রতিরোধ। এটি কলকাতার মেডিকা সুপার স্পেশালিটি হসপিটালের সাথে যুক্ত ডাক্তারবাবু ডক্টর সায়ক রয় মহাশয় পুঙ্খানুপুঙ্খভাবে বর্ণনা করবেন। এটি সন্ধ্যে 6:30 আরম্ভ হবে। Both the lectures will be very much helpful to us because the first one will give us a detailed molecular information about the main feature behind this pandemic and provide us a general guideline. In this, after the end of today's session, a feedback link will be provided to all the participants. I request all the participants to fill the feedback pro forma given in the link within the given time. I repeat in Bengali, আগের সেশনের শেষে সকল অংশগ্রহণকারীদের একটি ফিডব্যাক লিংক পাঠানো হবে। আন্তরিকভাবে অনুরোধ করব যাতে আপনারা ফিডব্যাক লিংক আমাদের পাঠানো ফিডব্যাক প্রো ফর্মাটি নির্দিষ্ট সময়ে পূরণ করে আমাদেরকে পাঠিয়ে দিন। Our chief patron, our honorable principal sir, Samanto sir, to say a few words. Sir, say a few words about our webinar.
Okay. Audible.
Sir.
Yes.
Yes, sir.
Yes,
Yes,
sir.
sir. Okay. Very good afternoon to all of you. Welcome to my dear friends, my fellow colleagues of Kalna College, friends of today's webinar. Firstly, today's and tomorrow's eminent speaker of this webinar, First of all, I welcome Dr. Sudit Sekhar Mukhopadhyay, Professor, Department of Biotechnology, NIT Durgapur, Dr. Sayok Rai, Consultant, Medical Hospital, Kolkata, Akesh Kundu, Assistant Professor, Department of Zoology, Vishwabharati, Professor Sudip Kumar Shil, Department of Botany, Professor of Botany, University. I welcome, sir, all of you participation here to deliver your lecture on today and tomorrow. Friends, I have already known that the topic discussed here is molecular aspects of and its possible management. Perhaps all of you know that what is molecular? Molecular is nothing but properties related with a particular molecule is called molecular. The molecule of COVID-19, that means RNA virus, and its activities and its associated related obviously will be discussed by our eminent speaker. I shall not say anything about COVID-19 and its related management, but rather I will say something about which is too much relevant here is science, scientific temper, and related issues. This is important. Perhaps all of you know that scientific temper, it is such a thing which cannot be developed automatically just by reading and writing science. Automatically, if you read and write science, it has to be developed, to be acquired by your attitude and by your practice. That is important. To follow the science, to follow the scientific method, to follow the scientific temper, it has to be acquired by a person. Yeah, that's why I will say a few words about science-related issues. That scientific temper, how can you acquire this scientific temper? Okay, how can you acquire? So science has its own method by which you can develop or you can acquire the scientific temper. Nothing but the simple method, experiment, observation, and inference. You have to be remembered here that you do not have to set up the laboratory apparatus, equipments, everything. Not that. Here you have to follow each and every aspect, the cause and effect of the ongoing system which you are following. That is important. The cause and effect of each and every aspect has to be followed and has to be analyzed by yourself and maintained throughout your life in each and every aspect. Then automatically you will see your scientific temper will be developed. Gradually developed. Remember here, conjugate cause, conjugate effect. You always will see there are so many conjugate cause and conjugate effect. You have to analyze those conjugate cause and conjugate effect. Logically, in science, there are some non-conjugate cause and non-conjugate effect. Perhaps all of you in your school life read Seebeck effect. In physics, all of you know the Seebeck effect. What is Seebeck effect? There is a conjugate cause and conjugate force. The conjugate force is that temperature gradient, and force is temperature gradient, and conjugate cause is, but there is a non-conjugate cause and non-conjugate. Non-conjugate cause is potential developed is automatically be generated, as well as due to potential difference generated, a current will flow. In your reality, you will always will see there are some, each and every aspect you will see there are some conjugate cause and conjugate effect. That means conjugate force and conjugate flux, as well as automatically a lots of non-conjugate cause and non-conjugate effects are there. So all the students, you have to be remember, you have to be sincere about all those non-conjugate phenomena which will be harmful to your life, So in each and every aspect, what you have to be done, you have to follow always positive conjugate cause and conjugate effects. You have to nourish always positive cause and positive conjugate cause and conjugate effect, and nullify all those non-conjugate causes and non-conjugate effects. These are the important things in science which we have to be followed in our daily life. That is important here. That's why I am telling you the everything related to the COVID-19, its molecular aspects and molecular managemental aspects. It will be discussed by our eminent speakers are here on today and tomorrow. So they will discuss everything to you, but I shall say these few words to my students. Always try to analyze yourself, the conjugate cause and conjugate effect, and nullify all those non-conjugate cause and non-conjugate effects, non-conjugate force and non-conjugate fluxes. I am telling you these things in Bengali, 2 or 3 sentences, and then I will stop my discussion. তুমি ভেবে দেখো আজকের এই পরিস্থিতিতে আমি যখন আলোচনা করছি তখন এই যে আলোচনাটা হচ্ছে এটা কখন হচ্ছে, এটা হচ্ছে বিকেল 5:30টার পরে। এবং কিভাবে হচ্ছে, যখন আমি ল্যাপটপের সামনে, আমার সামনে কোনো ছাত্র নেই, I am not in a position to transfer my emotion to my students or to my fellow participants who are listening my discussions. আমার কাছে কোনো সুযোগ নেই আমার এই যে বক্তব্যের আবেগটা তার কাছে পৌঁছে দেওয়ার। আমরা কি করছি, আমরা একটা অল্টারনেটিভ মিডিয়া, একটা ভার্চুয়াল স্পেসকে ব্যবহার করছি এখন, যে যা দিয়ে আমাদের এই আজকের আলোচনা, আমরা এই আজকের আলোচনাটা করছি কেন? আমাদের তো সিলেবাসে নেই এটা, ইট ইজ নট ইন আওয়ার সিলেবাস, আমাদের তো সিলেবাসে নেই এটা। আমরা আলোচনাটা করছি এই জন্য যে এটা আজকের দিনের প্রয়োজনে, আজকের সময়ে অত্যন্ত প্রাসঙ্গিক। ইট ইজ needful to the day for discussion. It is whether it is in our syllabus or not in our syllabus, but we believe that this type of discussion is essential for our society, for our student community. That's why we are discussing this topic in today. So like that, in your life, when you follow the science, when you follow in each and every aspect, you have to know what is the basic science behind it. Always you have to try to analyze, is there any science or not? Always in that way. And if you really go in that way, then you will see that your method of learning, it will turn to become different. তুমি নিজেই লক্ষ্য করে দেখবে তোমার মধ্যে যখন ধীরে 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 এই জিনিসটা, অর্থাৎ প্রত্যেকটা জিনিসের ভেতরে সত্যি তার কার্যকারণ সম্পর্ক, আমি বলেইছি যে কোনো একটা ঘটনার পিছনে একাধিক কার্যকারণ থাকে, তার মধ্যে কতকগুলো কার্যকারণ আমি বিজ্ঞানের উদাহরণ দিয়েই বললাম যে কতকগুলো কার্যকারণ থাকে, সেগুলোকে বলা হয় সরাসরি, যেটা করছি তারই বিপরীত প্রতিক্রিয়াটা দেখতে পাওয়া যায়। আর কতকগুলো পার্শ্ব প্রতিক্রিয়া, যেগুলো নিয়ে আজকে এখানে আলোচনা হবে এই কোভিড-19 আলোচনার সময়। আমার কথা হচ্ছে যে তোমার যে কোনো বিষয়ে, তুমি সায়েন্স পড়াশোনা করছো কি তুমি কমার্স পড়াশোনা করছো, এখানে কমার্স পড়তে গিয়ে তোমার যেমন কমার্সের প্রতি আগ্রহ জন্মাবে বা সায়েন্স পড়তে গিয়ে প্রতি আগ্রহ জন্মানোটাও যেমন স্বাভাবিক, তেমনি বহু পার্শ্ব ফেনোমেনা যেগুলো থাকে যা তোমাকে এখান থেকে অন্য পথে বিপথে চালিত করতে পারে, ডাইভার্ট করতে পারে। সায়েন্সে আমরা এগুলোকে বলে থাকি নন কনজুগেট ফোর্সেস, এবং ওই যে তুমি বিপথে চলে যাচ্ছ ওটাকে বলা হয় নন কনজুগেট ফ্লো, অর্থাৎ তুমি বিপথ চালিত হয়ে গেলে। সুতরাং আমরা যদি আমাদের এই যে সায়েন্টিফিক টেম্পারামেন্ট ডেভেলপমেন্টের জন্য আমরা যদি অলওয়েজ সমস্ত আমাদের কার্যকরী অবস্থায় থাকাকালীন আমাদের সমস্ত ক্রিয়াকলাপের পিছনে আমরা যদি কনজুগেট ফোর্স এবং কনজুগেট ফ্লাক্স এবং নন কনজুগেট ফোর্স অ্যান্ড নন কনজুগেট ফ্লাক্স তাকে অ্যানালাইজ করতে পারি, এবং ডিপেন্ডিং আপন দি অ্যানালিসিস অফ দি কনজুগেট ফোর্স অ্যান্ড কনজুগেট ফ্লাক্স অ্যান্ড non-conjugate force and non-conjugate fluxes, and if we try to nullify in every aspects of developing non-conjugate forces and non-conjugate fluxes, then we will go to our destination safely. And that is the important here on today's discussion. I shall try to request all the participants who are present here, they try to give your patience. Try to give your patience throughout the discussions. We have 4 eminent speakers. They will try to discuss everything in every aspect. According to my discussion, conjugate and non-conjugate way of cause and effects, everything will be discussed by them, and you can clear yourself. You can clear yourself by raising question to our speaker, and I think all of you who are present here, you will be satisfied. With these few words, I again congratulate to all of you. At present, you are 169 at present. This is at about 5:54. So remember that in real situation, there is no one in the college, only the principal. But here, in this classroom, there are 170 participants is here. So it is not a matter of joke. So this is a beautiful platform where we can share our opinion. So I request all the participants, you will participate wholeheartedly with our speaker and help them to complete the discussion. With these few words, I again congratulate to all of you for your patience hearing to me and for your such attendance on today's discussion. Thanking you all.
Thank you, sir, for your kind and stimulating words. We are grateful for the time and effort you took to share your views and opinion with us. Thank you, sir, for such inspiring words. Thank you. Now, I once again request all my participants to please put your audio and video mute or off and do not share your screen during the entire session. Now, I would like to introduce our first speaker of today's session, Dr. Sudhir Shekhar Mukhopadhyay. He is professor in Department of Biotechnology, NIT Durgapur. The topic that he will deliver today is COVID-19, the molecular. He pursued his master's in zoology from the Botany University and completed PhD in biochemistry from Bose Institute under Jadavpur University. He pursued his postdoc from Department of Molecular and Cellular Biology, Baylor College of Medicine, Texas, USA. After completing, he joined as a faculty in Department of Pediatrics, Texas Children's Hospital, Baylor College of Medicine, Texas, USA. After that, he joined as a faculty member in the Department of Molecular Genetics, MD Cancer Center, Houston, Texas, USA. In 2009, he joined as an associate professor in Department of Molecular Biology and Biotechnology in Tezpur Central University, Assam. And 2010, he joined as an associate professor in Department of Biotechnology, National Institute of Technology, Durgapur, and pursuing there as professor. He has many renowned publications in reputed national and international journals. Under his guidance, 4 scholars are pursuing their PhD. The current areas of his research interest are molecular mechanism of genomic instability and cancer, bioethanol, alternate renewable energy. Now I cordially invited Sir on the behalf of Kalna College to please deliver your talk. It over to you, sir, please.
থ্যাংক ইউ। ফার্স্টে আমি একটুখানি জিজ্ঞেস করছি আপনাদের কাছে, আমাকে বলা হয়েছিল বাংলাতে, তো মানে হোয়াট এভার ইউ হ্যাভ স্টার্টেড উইথ দ্য ইংলিশ, সো শুড আই স্পিক ইন ইংলিশ অর শুড আই স্পিক ইন বেঙ্গলি, হোয়াট এভার ইউ ডিমান্ড।
Sir, whatever your wish.
I don't have. I have.
আপনার যেটা সুবিধা স্যার, আপনার যেটা সুবিধা।
সুবিধাটা এখন এখানে আমার নয়, এখানে হচ্ছে ছাত্রছাত্রীদের অডিয়েন্সের কি সুবিধা সেটা যদি আমাকে বলা হয়, আই ক্যান মুভ। ওকে,
আমার বাংলা হলেই ভালো হয়, বেশিরভাগ বাংলাই বলছি।
ঠিক আছে, তাহলে আমি একটু স্ক্রিনটা সেট করে নিই, যদি আমাকে একটু হেল্প করা হয়। এই তো স্ক্রিনটা আমি সেট করে নিই।
ওকে স্যার।
হ্যাঁ স্যার, করুন।
হ্যাঁ, you are presenting your screen লেখা আছে, বাট কোথায় যাব? Now how to start?
সেটা দেখা যাচ্ছে কি? হ্যাঁ স্যার, দেখা যাচ্ছে।
আচ্ছা, ফুল স্ক্রিন দেখা যাচ্ছে? ঠিক আছে, আমি
ফুল স্ক্রিন দেখা যাচ্ছে।
স্ক্রিন করে দেন স্যার।
হ্যাঁ,
ফুল স্ক্রিন দেখা যাচ্ছে না স্যার, ফুল স্ক্রিন করে দিন।
দেখা যাচ্ছে না। আচ্ছা, এবার আমি করব।
এটা মিনিমাইজ করে দিন। হ্যাঁ, স্লাইডটা শুধু আপনি ফুল ইয়ে করে দিন, ফুল ইয়ে করে দিন।
এটা কি ফুল স্ক্রিন এসেছে?
হ্যাঁ স্যার।
হ্যাঁ স্যার, এসেছে। ওকে স্যার,
বাংলাতেই বলি। শুভ সন্ধ্যা। প্রথমেই আমি ধন্যবাদ জানাই কালনা কলেজের প্রিন্সিপাল এবং সমস্ত অধ্যাপক অধ্যাপিকা বৃন্দের, এবং বিশেষ করে আমার ছাত্র সম চন্দন আমাকে ইনভাইট করার জন্য এই সুন্দর একটা ওয়েবিনার সিরিজে, এবং খুবই এটা গুরুত্বপূর্ণ সময়। আমরা গত 5 মাস ধরে একটা অস্বাভাবিক পরিস্থিতির মধ্যে দিয়ে যাচ্ছি, যা আমরা সারা পৃথিবীতে এরকম একটা পরিস্থিতির সম্মুখীন কোনোদিনও হইনি। বা এই সম্পর্কে আমাদের অভিজ্ঞতা নেই, যদিও 1918 সালে এরকম একটা অবস্থা পৃথিবীতে হয়েছিল। তো প্রথমেই আমি বলে রাখি যে এই যখন আমাকে বলা হয়েছিল, মানে এর আগে আমি এই বিষয়ের উপরেও আরেকটি ইউনিভার্সিটি, কলকাতার একটি ইউনিভার্সিটিতে আমাকে টক দিতে বলা হয়, এবং তারপরে চন্দন মনে হয় ওই টকটা শোনার পরে আমাকে বলেন যে স্যার আপনি যদি দেন। তো আমি প্রথমেই বলে রাখি, শিক্ষার একটা বড় অংশ হচ্ছে নিজের লিমিটেশনসটাকে জানতে শেখা, যে আমার সীমাবদ্ধতাটাকে বুঝতে শেখা, বা আমি কতটা জানি সেই সম্পর্কে ধারণাটা। তো স্বাভাবিকভাবেই আমি নিজেকে মনে করি যে আমি নিজে একজন ভাইরোলজিস্ট নই। আমি মনে করি যে এই সাবজেক্টের পক্ষে বলার জন্য আমি উপযুক্ত নই, কারণ আমি নিজে ভাইরোলজিস্ট নই। এবং ইন্ট্রোডাকশনে আপনারা সবাই শুনেছেন যে আমি সাধারণত হিউম্যান জেনেটিক্সের উপরে কাজ করি, এবং হিউম্যান ক্যান্সার নিয়ে, সেল সাইকেল, বিভিন্ন অ্যাসপেক্টসে হিউম্যান জেনেটিক ডিসঅর্ডারের উপর আমি কাজ করি। এবং সুতরাং ভাইরোলজি আমার বিষয় না, যদিও আমার ভাইরাস ল্যাবরেটরিতে প্রতিনিয়ত আমাদের লাগে, সেটা অন্য ব্যাপারে লাগে, জিনকে ঢোকাতে সেলের মধ্যে লাগে। তো আমার তখন দুটো ভয়ের আমার কারণ আমার মধ্যে উদ্বেগ হয়েছিল যে এইটা সম্পর্কে বলতে গেলে। একটা হচ্ছে যে আমি নিজে এই ব্যাপারটায় অতটাও ভাইরোলজিস্ট, যেহেতু আমি ভাইরোলজিস্ট নই, সেহেতু আমি এই ব্যাপারটা অতটা সরগত নই। আর দ্বিতীয় ভয় যেটা ছিল, যে গত 5 মাস ধরে সোশ্যাল মিডিয়াতে এই ভাইরাসটা সম্পর্কে বা কাগজে এত কিছু ইনফরমেশন আছে যে আমি মনে করি সারা পৃথিবীর লোক অনেক বেশি জানে না এই ব্যাপারটাতে। তবে যখন আমি দেখলাম যে না, যখন আমাকে বারবার রিকোয়েস্ট করা হলো, তখন আমি মনে করলাম যে না, আমার একটা কোথাও একটা মানে রেসপন্সিবিলিটি এসে যায় যে কিছু লোককে বলার। সে বলাটা কি না, নিয়মিত যে সোশ্যাল মিডিয়াতে যেসবগুলো বেরোয় সেই খবরগুলো কতটা সত্যি, কারণ এই খবরগুলো অনেকটাই মানুষের মধ্যে কিছুটা মিথ এবং কিছুটা ফ্যাক্ট, সব তো ফ্যাক্ট নয় এবং সব তো মিথও নয়। এবং আমি দেখেছি আমার চারিপাশে অনেক ভয়ের উদ্রেক সৃষ্টি করে মানুষের মধ্যে, বিশেষ করে যারা বয়স্ক আছেন তারা অনেক ভয় সৃষ্টি করেন, এবং তারা অনেক কিছু ইতিমধ্যে মিডিয়ার থেকে কিছু কথা শুনে যেসব বাড়িতে প্র্যাকটিস করেন, যেসব খান, আমার মনে হয় সেটা করা উচিত নয়। সেই অ্যাসপেক্টসে আমার মনে হয়েছিল যে কিছু একটা যদি করতে পারি। তো সেটা করার জন্য আমি যেটা করেছিলাম, যেহেতু আমরা বৈজ্ঞানিক, আমরা সবসময়তেই প্রত্যেক সপ্তাহে পৃথিবীর যত বড় বড় জার্নালগুলো বেরোয়, নেচার, সেল সায়েন্স, নিউক্লিক অ্যাসিড রিসার্চ, এবং এইসব জার্নাল বা নিউ ইংল্যান্ড জার্নাল অফ মেডিসিন, এইগুলো নিয়ে আমি দেখি যে এইগুলোতে কি কি ধরনের পাবলিকেশন হয়েছে এই কোভিড-2 এই ভাইরাসের ওপরে, এবং সেইগুলো এর সাথে WHO, ওয়ার্ল্ড হেলথ অর্গানাইজেশন বা সিডিসি অ্যাটালান্টা, যেটা ভারত পৃথিবীতে ওয়ান অফ দ্য ইনফেকশাস ডিজিজের জন্য অন্যতম, তো তারা কি কি তারা পাবলিশ করেছে, সেই ডেটাগুলোকে নিয়ে আমি বলতে চাই যে কোনটা ফ্যাক্ট এবং কোনটা মিথ। সেটাই আজকের আমার কথা, এই টকের মেন উদ্দেশ্য। তো আমি সবসময়, যদিও এটাতে ইন্টারাকশন সামনাসামনি হচ্ছে না, তো প্রশ্ন আপনাদের থেকেই যাবে, কিছু কিছু প্রশ্ন যেগুলো টকের পরে অবশ্যই আপনারা প্রশ্নগুলো করবেন, আমি যতটা পারবো আমার জ্ঞানের তরফ থেকে আমি উত্তর দেবার চেষ্টা করব। আচ্ছা, প্রথমেই বলে রাখি যে হোয়াট ইজ ভাইরাস, ভাইরাসটা কি। যদিও বায়োলজির ছেলেরা এটা সম্পর্কে ভালো অভিজ্ঞতা আছে। তো ভাইরাস কি একটা জীব, না ভাইরাস একটা জ্বর? জীব বা জ্বরের পার্থক্যটা কি? আমরা জানি যে জীবের দুটো মেন ক্যারেক্টারিস্টিক্স, একটা হচ্ছে তাকে বংশবৃদ্ধি করা, রিপ্রোডাকশন, আর একটা হচ্ছে মেটাবলিজম। মেটাবলিজম মানে জীব তার খায় যে ফুডটাকে মেটাবলাইজ করে, এবং তার মধ্যে যে পোটেনশিয়াল এনার্জি সেটাকে সে কাজে লাগায়, বংশবৃদ্ধির কাজে লাগায়। তাহলে ভাইরাস একটা, আমরা বলি আমাদের ভাষায় ভাইরাস একটা পার্টিকেল, ভাইরাসের মেটাবলিজম নেই। ভাইরাস হচ্ছে বৃদ্ধি করতে পারে, সেই বংশবৃদ্ধিটা ভাইরাসের অন্যভাবে, সেটা আমি পরে ডিসকাশন করবো ভাইরাস কিভাবে বংশবৃদ্ধি করে, কারণ স্ট্রাগল ফর দ্য এক্সিস্টেন্স, এটার জন্য পৃথিবীতে সব যত রকম মানে জীবের বা উৎপন্ন যারা যারা এখানে ইভলভ করেছেন, প্রত্যেকে এখানে এক্সিস্ট করতে চায়, তো তার জন্যও ভাইরাসও নিজেকে রেপ্লিকেট করতে চায় এবং এই পৃথিবীতে এক্সিস্ট করতে চায়। কিভাবে এক্সিস্ট করে বা পৃথিবীতে কিভাবে নিজেকে রেপ্লিকেট করে সেটা আমি বলবো। প্রথমেই আমরা জেনে নিই তাহলে ভাইরাসটা কি। ভাইরাস, আমরা এটাকে যদি ভাইরাস দেখি, আমরা বলি ভাইরাস একটা প্লুরাল শব্দ এবং সিঙ্গেল আমরা বলি ভিরিয়ান। তো ভাইরাসের যদি আমরা স্ট্রাকচারটা দেখি, প্রথমেই ভাইরাসের হচ্ছে যে আমরা দেখি যে ভাইরাসের একটা নিউক্লিক অ্যাসিড দু রকমের আমরা জানি, আরএনএ বা ডিএনএ। কোনো ভাইরাস আরএনএ ভাইরাস, কোনো ভাইরাস ডিএনএ ভাইরাস। এবং তারপরে এই নিউক্লিয়াসের পরে ভাইরাসের একটা প্রোটিনের আবরণ দেখা যায়। বেসিক্যালি ভাইরাস হচ্ছে প্রোটিন এবং নিউক্লিক অ্যাসিড নিয়ে তৈরি। এই প্রোটিনের আবরণটাকে আমরা বলি ক্যাপসিড। আর তারপরে যে একটা আবরণ আছে এবং তার মধ্যে অনেকগুলো স্ট্রাকচার আছে, এইগুলো হচ্ছে ভাইরাসের একটা ভিরিয়ানের একদম বাইরের আবরণ। এবং এইগুলো বিভিন্ন ধরনের প্রোটিন স্ট্রাকচার থাকে, যেটা ভাইরাসকে নিজের যে হোস্ট, সেই হোস্টকে চিনতে সাহায্য করে বা হোস্টের মধ্যে ঢুকতে সাহায্য করে। এটা যে সিম্পল। তাহলে ভাইরাস কি দাঁড়ালো? ভাইরাসের তার শরীরটা তৈরি হচ্ছে নিউক্লিক অ্যাসিডস এবং প্রোটিন দিয়ে তৈরি। ওকে, এইবার যদি আমরা নেক্সট স্লাইডে যাই তাহলে দেখি কিভাবে ভাইরাস রেপ্লিকেট করে। আমি আগেই বলেছি ভাইরাসের একদম বাইরের দিকে যে প্রোটিন থাকে, সেই প্রোটিনের সাহায্যে ভাইরাস হোস্টের মেমব্রেনের কোন একটা প্রোটিনকে সে রিকগনাইজ করে। আমরা এটাকে বলি রিসেপ্টর এবং এটাকে যদি লাইগ্যান্ড ভাবি, তাহলে কি হয়? প্রত্যেকটা ভাইরাস তার নিজস্ব রিসেপ্টর উপরে বাইন্ড করে এবং আমরা বলি হোস্ট রিসেপ্টর এবং লাইগ্যান্ড ইন্টারাকশন। তারপরে ভাইরাস কি করে? তার এন্ডোজোম আমরা বলি, তারপরে সেলের মধ্যে ভাইরাসের তারপরে সে ভাইরাসের সেলের মধ্যে ঢোকে। আলটিমেটলি ঢোকার পর ভাইরাস যে নিউক্লিক অ্যাসিডস আছে, যা ডিএনএ অথবা আরএনএ, এর মধ্যে কি থাকে? এর মধ্যে ভাইরাসের জিনগুলো থাকে। কি জিন? না, ভাইরাসের স্ট্রাকচারের জন্য যে প্রোটিন দরকার, সেই প্রোটিনগুলো এখানে থাকে। তো ভাইরাস প্রথমেই হোস্টের মধ্যে ঢোকার পর এর মেন উদ্দেশ্য হচ্ছে এই জিনগুলোকে অন করা। কিভাবে? তো জিনগুলোকে ট্রান্সক্রিপশন এবং ট্রান্সলেশনের মাধ্যমে প্রোটিন তৈরি করে। সেই প্রোটিন তৈরি করার যে মেশিনারিজ লাগে, সেগুলো সমস্ত সে হোস্টের থেকে নেয় এবং তারপরে সে কি করে? তা প্রোটিন তৈরি করে। ইতিমধ্যে সে তার নিউক্লিক অ্যাসিডটাও তৈরি করে এবং আরএনএ এবং নিউক্লিক অ্যাসিডস এবং প্রোটিনগুলো অ্যাসেম্বল করে অসংখ্য ভাইরাস তৈরি হয়। তারপর সে আবার তার হোস্ট ছেড়ে বেরিয়ে যায়। একে বলে এক্সোসাইটোসিস। সো এন্ডোসাইটোসিস, এক্সোসাইটোসিস। আমি আগেই বলেছি আমাদের দু ধরনের ভাইরাস আমরা পাই, একটা হচ্ছে আরএনএ ভাইরাস এন্ড ডিএনএ ভাইরাস। কিছু আরএনএ ভাইরাস, যেমন রেট্রো ভাইরাস, HIV ভাইরাস, এটা রেট্রো ভাইরাস, এরা আবার নিউক্লিয়াসের মধ্যে যায় এবং সেই আরএনএটাকে ডিএনএ বানায়। সেই ডিএনএটাকে সে হোস্টের ক্রোমোজোমের মধ্যে ঢুকিয়ে দেয় এবং সেই হোস্টের ক্রোমোজোমে ঢোকার পর সেখান থেকে সে ভাইরাস তৈরি করে বেরিয়ে যায়। কিন্তু এই ভাইরাসটা চিরকাল তার নিউক্লিয়াসের ক্রোমোজোমের মধ্যে রয়ে যায়। ওকে, এবং সবচেয়ে বড় কথা যেটা বলতে চাইছি, যে প্রত্যেকটা ভাইরাসের বাইরে কি থাকে? একটা নির্দিষ্ট প্রোটিন থাকে, যে প্রোটিনের সাহায্যে সে হোস্টের রিসেপ্টরকে রিকগনাইজ করে। এটা হচ্ছে ভীষণ স্পেসিফিক। এই হোস্ট রিসেপ্টর অ্যান্ড লাইগ্যান্ড ইন্টারাকশন ভীষণ স্পেসিফিক। প্রত্যেকটা ভাইরাস তার স্পেসিফিক হোস্ট আছে। প্রত্যেকটা ভাইরাসের, ইভেন ব্যাকটেরিয়ারও ভাইরাস আছে, আমরা বলি ব্যাকটেরিওফাজ। সেই ব্যাকটেরিওফাজ কিন্তু ব্যাকটেরিয়াকেই আক্রমণ করতে পারে, অন্য কাউকে আক্রমণ করে না। এবং সেইভাবেই প্রত্যেকটা এই পৃথিবীতে প্রত্যেকটা প্লান্টেরও ভাইরাস আছে। সেই প্লান্ট ভাইরাস প্লান্টকে, স্পেসিফিক প্লান্টকেই আক্রমণ করতে পারে, অন্য কাউকে করে না। কিন্তু পরের ইয়েতে পরের আমার অ্যানালিসিস আমি দেখাবো যে কিভাবে এই কোভিড ভাইরাসটা কিভাবে একটা হোস্ট থেকে আর একটা হোস্টে সে পরিবর্তন নিজেকে করেছে। তো প্রথমেই আমরা জেনে নিই তাহলে ভাইরাস হোস্টের স্পেসিফিক একটা রিসেপ্টরের উপরে বাইন্ড করে এবং এটা ভীষণ পরিমাণে স্পেসিফিক। এবং প্রত্যেকটা ভাইরাস তার স্পেসিফিক হোস্ট আছে এবং হোস্টের মধ্যে ঢুকে সে একটাই কাজ, সে নিজেকে বাড়িয়ে নেয় এবং বেরিয়ে চলে যায়। এবং কোনো ভাইরাস, যেমন রেট্রো ভাইরাসরা, তারা কি করে? হোস্টের ডিএনএ এর মধ্যেই অবস্থান করে। তো এই হচ্ছে ভাইরাস সম্পর্কে আমাদের ছোট্ট একটা অ্যানালিসিস। এরপরে আমরা চলে আসি
সরি টু ডিস্টার্ব ইউ স্যার। কয়েকজন চাইছে মানে তারা একটু বাংলা একটু বোঝে না, তাদের জন্য যদি আপনি কিছু টার্ম যদি একটু ইংলিশে ইউজ করেন। দেয়ার আর সেভারাল পার্টিসিপেন্টস, তার চ্যাট বক্সে লিখছে যে ইংলিশ প্লিজ ডোন্ট আন্ডারস্ট্যান্ড বেঙ্গলি ল্যাঙ্গুয়েজ এরকম কয়েকজন লিখছে, সেই জন্য মাঝে আপনাকে
I thought probably it's better to. Acha, can you please tell me that is it very difficult to both the the English and Bengali? Either you can ask me to speak the English or then ask me to speak Bengali because কি হয় জানো তো মানে আমি যখন বাংলায় বলছি নিজেকে মানে না আমি বুঝতে পারছি না ওরা কি চাইছে যে পুরোটা ইংরেজিতে বলি, একটু দেখে নাও।
সেটাই মানে স্যার কেউ কেউ বলছে ইংলিশ, কেউ কেউ বলছে বেঙ্গলি, আর আমাদের টাইমেরও একটা কনস্ট্রেন্ট আছে, সেটা তো অবশ্যই।
আচ্ছা ঠিক আছে, আমি দেখছি কতটা
ইংলিশে বলুন, স্যার, ইংলিশে বলছে সবাই।
Well, that's very good. Okay, let's go ahead.
Thank you, sir.
Okay, so should I repeat the first slides or is it okay with you guys? So should I repeat the first slides? What is virus and how virus replicates? So is it okay? So then let's move to the SARS virus. So that is the today's topics. So
Yes,
why
sir. Start from here, sir.
So English is okay? ঠিক আছে Chandan?
Yes, sir. একদম ঠিক আছে।
Okay, so now why it is called the SARS? So it is why SARS is all you know that it is a basically respiratory syndromes. But why it is called the COVID? COVID, the words comes from the corona. Why it is called the COVID corona? Why does it call the corona? So if you know that there are two, now I found in the literature there are two, that there are two hypotheses there. So the corona, so that's called the corona. The word came from the crown, and then they also said there are some systems during the solar eclipse. So when the moon's shadow covers all of the the sun, and then that time some of the sun's rays radiates, and that system's called the corona. So these two things has a resemblance with this virus's structures. That's why it's called the coronavirus. But so now the virus is a COVID-2, coronavirus 2, and if you know the same, I told you the coronavirus structure is same like I discussed you. This is a central nucleic acids. Now in case of the COVID, it has a RNA, and then it's a that's called the inner membrane, and then this is the outer membrane, and these outer proteins, and these are the proteins, basically dumbbell-like proteins. These are basically called the spike proteins, which is the most important proteins of the virus, the important part of the virus. That's with the spike proteins, they interact with the host and enters the host cells. So now go to the next slides. Now, so is it? We just call the novel. So is it really novel? Novel means it's a totally new. Nobody knew about this virus, coronaviruses. There are different types of viruses. So corona is the one kind of viruses. Before that one, whatever the coronaviruses have evolved in this earth, they have been classified alpha, beta, and delta and gamma. But basically India, we found alpha and beta. But right now, is it really the new 2019 or 2020, this virus, is it really the new virus? No. If you see the data, 1960, the first human coronavirus OC43 evolved, and then 1960 there are two, another one human COV229E, and then I found that 2003 there is another attack of epidemic of human coronavirus, that is the coronavirus beta, and 2005 and 2012 and 2019. But if you see, so it's not all on a sudden this coronavirus become epidemic in this earth 2019 2020. So it started from the 1960. But the objective of my slides, if you see the symptoms associated with the coughs, if you see the most symptoms is the fever, cough, upper respiratory symptoms, there is a common symptoms, and then flu-like symptoms. These are common symptoms all of the coronaviruses. But now one of the very much people are concerned is called the spreader. Now this is very infectious. It is a highly infectious the particle. So there is a term called the super spreader. Now I'm trying to compare what is the super spreader and what is the normal spreader. So if you see the slides, there is a one person, he is an infected person. So he directly infect one person, and then this person can infect another one. So this is a direct infection, this is an indirect infection. So this is a very common. just like a flu virus is a very common, this kind of spreads it's called the flu virus. And what is super spreader? But this is the infected persons. At a time, he can spread many people together. It's called the super spreader. Now, think about the coronavirus. What is it? What kind of the infection spreads? So now I'm talking about, first come to the 2003. I'm showing you some of the recent cases. 2003. So there is one doctors with the SARS virus in Hong Kong. So there is one doctor infected with the SARS virus 2003, and he infected almost 100 plus people, and he himself died. 2015, patients with South Korea with MERS. I will tell you what is MERS. So MERS is another, it's a Middle East virus. It's the same coronavirus. It's evolved in the Middle East. So one patient in South Korea was attacked with the MERS virus, and he died. But before he died, so he spreaded almost 82, the persons. But in 2020, the COVID-19 or COVID-2, what is that? You see that this is the infected person. He can infect 11 people. So think about, compared to that, whatever the others epidemics happen due to the COVID virus in this earth, compared with this today's COVID-2 viruses. But whatever the social media is not saying that one so quickly. So now I'm giving you another example. What is that one? How contagious is COVID-19? Because there's a lots of rumors spreaded all over that the social media. So now if you see average number of people infected by an individual, we call the RG value. Average number of people infected by an individual. So think about the measles. Measles is another virus related disease. Its RG value is 12 to 18. That means it can infect 12 to 18 people at a time. Smallpox 5 to 7, polio 5 to 7, mumps 4 to 7, HIV 2 to 5, and then influenza 2 to 3, SARS-CoV, the earlier SARS-CoV 2013 and 2002 2 to 5, and Ebola 1.5 2.5, whereas COVID-19 1.5 to 2.5. So compared to the other viruses, if you see the infection ability, it's not that much whatever the social media is talking about. Okay, now again I'm showing you another example how COVID-19 compares to other major viruses. So what are the other major viruses? If you see, so I'm not talking about what kind of virus, but these are all the made the epidemics in this earth: Mark virus, Ebola, and all this virus. So now they have been made epidemics in this earth different years. But I like to concentrate you only in this area, the fatality rate. That means the death rate due to this infection. If you see Mark virus, 80% fatality rate, Ebola 40%. Hendra 57, H1N1, the bird flu 52%, Nipah 77%, SARS-CoV, that means earlier SARS-CoV 9.6%, H1N1 17%, MERS-CoV, that means I told you that coronavirus evolved in the Middle East 34%, and H7N1 bird flu 39.3%, but COVID-19 3.38. If you see today's India's data, it is less than 3%, 2.28 fatality rate. So we should not be worried that much about the COVID-2. Now, I am showing you another very interesting comparison, these viruses. So now there are 3 viruses I'm showing you. One is the MERS. So MERS is, I told you again, that's the MERS virus evolved in the Middle East respiratory syndrome. And this is the SARS 2002, earlier SARS 2002 and 2004. This is also COVID, but these are all COVID viruses. And today's SARS-CoV-2. So now just go to the last line. Go to the last line of the table. Ventilator use. So in case of MERS, 24.5% ventilator was used. In case of SARS-CoV, earlier CoVs 14 to 20%, and then SARS-CoV-2 4.1%. Think about what the media is saying, what the social media is saying, that everybody is now very freaky, or everybody is now become frightened because they thought probably if you get this disease, you have to have the ventilation systems, you have to go to the very, the delicate hospitals, otherwise you won't survive. You need a ventilator. But this is a myth, if you see, compared to the other viruses. Now, come to that very important, the analysis of this stock, analysis of the stock. What is that one? Here are two pictures I'm showing you, left and right. So left, this is a Hunan, the meat market. And if you see, there's a lots of bats. Actually, the bats are, they are selling in this Hunan meat market and the other types of meats market. By the way, I'm telling you, the whatever the earlier SARS, so SARS 2 and SARS, the 2002 and then 2009, whatever the SARS was evolved, both the SARS was evolved from the meat market of the China. So again, this SARS virus also evolved from this Hunan. There is a controversy. It came from the Hunan meat market, and there is another Institute of Virology. They were also studying on the bats COVID virus. So people have a the controversy whether this is evolved, this released from this institute, or it's released from this meat market, because lots of bats are selling here, and there is already the evidences are there. This kind of birds are evolved from the meat market. So now another controversy came. This is very much the controversy that it was be man-made or it was generated in the Wuhan Institute of the Virology. So in my next slides, couple of slides, I will try to analyze this answer with that analysis of this genome of this COVID virus too. So now I'm not going to that all the details. So now what the scientists did, how did the scientists they identified where it came from, where the origin. So now they have collected all these viruses samples and then sequenced all the genomes, and then they find out the similarity between the genome. And from this analysis, the hypothesis have came out, the anonymous hypothesis. Actually, this virus evolved from the bat, and there are some intermediate hosts. What are this? In case of SARS and SADS, there are some intermediate cat, pig, and then in case of MARS, this is a camel, and then in case of SARS-CoV-2, so hopefully I will tell you in my later slides, it is a pangolin. It is an anteater pangolin. Okay, so originally it's evolved from the bat, and from bat, before the human attack, there are some secondary, the host, and from there it came to that host, came to that human beings. So this is the analysis. This is the analysis done by the scientists, and then how did they do? They have sequenced the genome and then analyzes the homology. How much homology from these viruses to the bat viruses as well as the other animal viruses, and from this homology search they have come to these conclusions. Now again I'm telling you, now I'm going to that, showing you this is the genome analysis of the SARS-CoV-2. So now it is a RNA virus, and then whole RNA virus, 30,000 base pair. This consists of the 30,000 base pair RNA, and this whole RNA, on the basis of the gene expressions, we can classify it into ORF1. ORF means open reading frame. Open reading frame, that means the open reading frame means it has the 3 nucleotides make 1 amino acids, and many amino acids make a 1 proteins. So now if it is a frame, that means that specifically 1 amino acids make the another amino acids, and then another one. But if it is a frame changed, so automatically there will be no expressions of the amino acids. So that's why I call the open reading frame. So now open reading frame 1B, 1A, 1B, and ORF 2 to 10. So now just think about this blue ORF and the red ORF. Basically we call the replicase genes. Why it is called the replicase? From these sequences, whatever the proteins are formed, these are the virus own proteins. So these proteins basically they help to produce that virus structure and protein. That's why we call the replicase protein. So replicase genes, what they do, they basically synthesize the virus structural protein as well as the virus's RNA or nucleic acid, whatever here is the nucleic acid, the RNA. So hopefully the replicase genes are involved in the the production, replication of the RNA as well as the other virus structure proteins. So now ORF 2 to 10 So green one, if you see, there are a couple of genes are there, and all these proteins are basically structured proteins. Whatever I showed you my earlier picture, the outside structure, the different structural proteins are basically generated from these regions of this RNA. So this is the virus genomes. Now, before going to the analysis of the virus genome, I will try to give the answer how it is originated. It is a man-made from the analysis of the genome. Before going to that one, I like to show you the life cycles of the SARS-CoV-2. So already I told you the virus have a virus. This is the nucleic acids, and then outside, and these proteins are the virus ligand. Ligands here is the spike proteins. So the spike proteins interact with the human cells specific proteins called the ACE2, angiotensin converting 2. It's a receptor, angiotensin converting 2. So receptor present in the membrane. So right now, that's why the spike protein first recognize the ACE2 proteins and ACE2 proteins, and then they enters the human cell. But why, so then when they first interact with the ACE2, there is one protease called the transmembrane protease. When they interact, this ligand interacts with that ACE2, this transmembrane protease degrades, and as a result, whole virus is fused with the human cell membrane, and ultimately they enters the human cell. This is called the endocytosis. There are lots of proteins are involved. There are mechanisms. I'm not going to this is not that part of this, the talk. But anyway, with the first, the ligand protein, here is the spike protein, recognize the ACE2 receptors of the human, and then with the endocytosis, they enters. But if you see, so why it is called the respiratory syndrome? The ACE2 proteins, angiotensin converting enzyme 2. I'm just telling you very briefly. So we have a protein called the angiotensin. So angiotensin is evolved from the angiotensinogen. Angiotensinogen to angiotensin converted by two enzymes called the ACE2. That means angiotensin converting enzyme 1. Another one, angiotensin converting enzyme 2. So then angiotensin come. What is angiotensin? Angiotensin and renin, there are two opposite actions of the molecules we have. One is called the vasodilator, another is called the vasoconstriction. That means they constrict our the alveolar systems, or they constrict or dilated our alveolar systems. That's why they control the pressure, blood pressure in our systems. So now ACE2 is mostly expressed in the nasal the nasal region. There's all these epithelial buccal epithelial cells and alveolar cells. That's why the virus first enters in our body through the mouth as well as through the nasal duct, and ultimately through the trachea. It's infected whole alveolar systems, and that's why respiratory systems, it's called the respiratory syndrome, and then causes the respiratory syndrome. So now, once it is entered into the cells, what is I told you, the virus RNA, first they generate the protein called the replicase, and then again replicase start the production of the protein, the structural protein, as well as the virus RNA. So now, when the structural proteins, virus RNA generated in the cells, then these two proteins become assembled in the takes part in the endoplasmic reticulums or in the the Golgi bodies, and then these proteins now with the protein assemble of the protein and nucleic acids. So lots of virion evolve, and then they get out. It's called the exocytosis. So this is in a brief, the SARS-CoV-2, how the SARS-CoV-2 infect us, and then life cycle in the cells. So this is basically the scientists are saying this incubation period is 1 to 14. But sometimes it's also increased. But basically, whatever the data I found, this life cycle is a 1 to 14 days. So now come to that main analysis part of this talk. What is I told you? So now I will try to analyze the gene, and with that help of the analysis, I will try to give you an answer how it is evolved. So now I'm not going to the analysis of the whole genome, but only I like to concentrate on the spike protein. Right now what is spike protein. So now this is the spike protein. So this is the elaborated spike protein. Spike protein can be divided into two parts. It's called the S1 part and S2 part. S1 part basically receptor binding domain. So the S1 part basically responsible for the ACE2 binding. Okay, and S2 domain basically gets infusion of the membrane and gets inside the inside the cells. So now first talk to the this domain, S1 and S2 domain. But before the S1 S2 domain, there is a one hinge region. So the left part is the S1 domain and right part is the S2 domain. So now, so the question is, so it was the bat virus. Basically, it is host was the bat. They attack the bat. They replicate in the bat's body. But how it's become to the changes is host, a secondary host pangolin, and then tertiary host in the human. How it has happened? So this is the questions today. Okay, now so virus, I told you there are every living beings in the earth. Our genome is always changing. So there are different we call the mutation. How it is changing? So there are different DNA damaging agents, and we have a repairing systems. So repair, it's not at 100%. So obviously some of the parts, some of the region is left without repairing, and that ultimately changes in the genome. That is called the variation, and that variation leads to the evolution. Evolution is the dynamic process. So hopefully this virus also under evolution, so they change their genome.
স্যার আপনার অডিও মিউট হয়ে গেছে। এক্সকিউজ মি স্যার, আপনার অডিও মিউট হয়ে
স্যার আপনার অডিও মিউট হয়ে গেছে স্যার। উদিত স্যার আপনার অডিও মিউট হয়ে গেছে।
Now it is okay.
Yes sir.
ওকে স্যার।
Okay.
Yes, sir.
Sorry, it has happened. Actually, it's also kind of change. Well, now it is okay.
Yes, sir.
Okay, now I'm audible. So now come to that S1 part. S1 part is a consist of two domain. One is called the N-terminal domain and the receptor binding domain. This is only S1, concentrate on the S1. So now think about, so this is the this is the amino acid sequence. It's a SARS-CoV-2 virus. The virus is a COVID-19, and this is a bat virus, original bat COVID virus. This is a pangolin. These are two types of pangolin. One is the GX, one the GD. Actually, they sell this pangolin in this Hunan market. They brought it from the two region of the two region of the China. So on the basis, this called the GX, Jiangsu and something like that. So pangolin, there are two types of pangolin and then bats. But concentrate on this region, amino acid region. If you see the bats, SARS-CoV-2, there is a 5 amino acids, Y, L, T, P, G, D. So Y, L, T, P, G, D, you do not find in the pangolin here, the pangolin GD. You find in the bat Y, L, T, P, G, D, exactly in the bat COVID virus, you find exactly these amino acids. But in case of pangolin, you see Y, L, T, P, G, there is no D. So see, so this is exactly the resemblance with that came from the bat, and then pangolin, actually they have only the missing the D, but they have all. But other than other COVID viruses, this sequence are not present. So this is a N-terminal domain. And now come to the receptor binding domain. So this come to the receptor binding domain. So now those who are that we represent the protein in a graphical pattern. So this is the way. So we call the graphical this is nothing but the representation of the protein AC2. This is the receptor. there's an angiotensin converting enzyme 2. And now that how this virus, this part, the RBD part, the receptor binding part interact with the AC2. So basically there are 6 amino acids are responsible for interaction of the AC2. 1, 2, 3, 4, 5, 6. If you see, and if you see again these amino acid sequences of all these viruses. So if you see, there are some unique. So SARS-CoV-2 have a lysine, bat has a lysine, and pangolin has a lysine. Other than they do not have a lysine. But if you see, this is the same. But again, this one is a 493 amino acid, aspartic acid only present in the COVID-2, and aspartic acid present in the pangolin, but not in the bat virus. So this is the way, if you see, so this is the way we predicted. So basically these sequences changes such a way, basically it was origin in the bat, and now it is at random changes, and as a change, first is attacked, it is attacked the pangolin virus, it is a pangolin virus, pangolin cov virus. So now ultimately they change such a way they can interact with the human AC2. So obviously, so from this analysis, I can assure you, I can confirm you. So how the variation, due to the variation or changes in the genome, so ultimately the bat virus changes its host from the bat to the pangolin, and pangolin to the human. But anyway, I just missed another thing. So now this is the 5 amino acids. Why the 5 amino acids, when they have integrated in this region, what the advantage of this protein? So now basically this is a protein structures, and if you see, basically it's a tetraprotein. Then 3 same proteins are conjugated together, and they are there. So now this trimeric, homotrimeric protein. So this is this part. The red part is basically these 5 amino acids. So now scientists are saying, one, it is changed into these amino acids. So this region become protruding. So protruding means once it's protruded, it helps to interact with the AC2. So that is the hypothesis made by the scientists. And I told you this receptor region, how it is evolved for the basically COVID, the bat COVID, and ultimately intermediate is the pangolin COVID, and ultimately it's become a human COVID, and that's why it starts interacting with the AC2, human AC2, and they can enter into the human host. Now come to that another very much challenging questions, how it become contagious. So to understand the contagiousness, I'm just trying to explain you by analyzing the S2 subunit. The spike protein S1 subunit, from I told you where it is originated. So where it is originated is man-made, or it is originated from the bat, or it is originated is the bat, or it is varied. So I told you, obviously these variations cannot make in the laboratory. Those who are working those who have experiences. So we do lots of mutations in our genome in the laboratory, but these variations, it's at random variations. We cannot make these variations in the laboratory. So hopefully, scientist hypothesis. This is not my hypothesis. This is scientist hypothesis in the paper. They said hopefully by natural variations, the bat origin came into the intermediate origin pangolin, and ultimately it changes its host in the human. So now the question is how become contagious. So I just try to concentrate you only this region of the S2 subunit. This is the middle region of S1 and S2. So if you see, these are all the sequences from the human CoV2, bat, pangolin, and other COVID-2. So now if you think there are RRAR. So RRAR, if you see the arginine, alanine, and arginine, these are polybasic sites which is only found in the human COVID-2, but it's not found in the any other COVID viruses. So only the bat has a arginine, the pangolin has a arginine. But if you see, these are the this is the unique sequences have been found in this region COVID-2. So it is called the polybasic, that this is the basic amino acids. But I will try to explain you as a result of this incorporation of the amino acid, what is the advantage, how it helps the virus into contagious. So before that one, I try to elaborate this region again. If you see PRRA, only the pangolin has AAR and P. Other than one, none of the COVID viruses, this amino acid is missing here. So this is a very unique for the COVID-2 change. Now come to that, what is the advantage of this incorporation of this amino acid? It's called, I told you, polybasic, the cleavage site. Purine, there is a protease called the furin. Basically, purine is a protease. Protease means what? Protease, protein degrading enzyme called the protease. I told you transmembrane protease when I described that cell cycle. Here is another protease called the furin. So purine, so this region, basically the furin protease cleavage site. So protease, specific amino acids, they binds and then cleaved. So that is called the protease cleaving site. So purine basically cleaves in this arginine A and R, this region. So when the COVID-2, as a COVID-2, this amino acids has incorporated and then purine cleavage site has generated. So what is the advantage of this purine cleavage site? So there are some evidences. So one of the evidences is the avian bird flu. So what the scientists did, actually avian bird flu, it is not that very contagious. So now there is one gene called the hemagglutinin esterase. So hemagglutinin esterase, so in this gene, so scientists, they incorporated this polybasic cleavage site, purine cleavage site, and they found this avian virus, which is not very contagious, it become contagious. But how it is contagious? There are a couple of hypotheses. Obviously, it is not, there lots of work has been done, but it is not confirmed, but it is a hypothesis. People are saying, scientists are saying, so when this, the sequence are generated, so before the whole virion coming out of the host cells, this protease clipped. And as a result of S1 and S2 protein is separated. And that separation helps the virus to and then fusion with the human protein after next infection. So that is the hypothesis actually have been made by the different scientists. They said % of the fusion protease sites, due to this incorporation, it's become contagious. So this is the information I have. So now I think I make you believe or strong believe. I don't know whether it's a strong or something, but at least I made you believe that this virus is where it is originated and how virus changes its host from one host to next host. But how it is virus changes their host? This is a very the hypothetical question. Some people says if the original host is become endemic or number of the original host, if you eat the bat and bat and bat, automatically there is a running out of bat. Obviously a struggle for the existence. This virus choose change their genome, choose another host. So another people saying, so when you are become the contaminations of this bat, so people are eating more and bat, automatically this virus, so they left the virus, changed their genome and started attacking to the human. So there are lots of hypotheses, but at least I believe that it is nothing but the variation of the genome. As a result of the variation of the genome, they change their host. So now come to another very important, the myth. So in the social media, before the summer start, very many people in the social media said, now summer is coming, so this virus will be burned. In some of the Australia and New Zealand, they said winter is coming, so virus will be frozen, so the epidemic will be stopped. But it has not happened. So I tried to understand, so is there any effect of the virus, the evolved or virus, due to the there is some relations with the climate. So now I'm talking about there are two things. One is the El Nino. All of you know the El Nino. El Nino basically the warm temperature, the alterations in the earth, basically the Pacific Ocean. And then La Nino is the opposite. La Nino is the cold temperature alterations. All of you know the El Nino effect, El Nino effect. So this is a warm temperature variations in the world, and La Nina is the opposite. There's the cold temperature variation in the world. So now I have found that in the one paper they have shown, so all these different virus are evolved either El Nino or La Nina. Either in the cold temperature variation, if you see the SARS, HQ2, COVID in the El Nino, they evolved during the El Nino, and some of them evolved during the the La Nina. Oh, sorry. Okay, so hopefully I can tell you. So obviously the temperature, there is no relations with the virus infection with the temperature, because some of the virus evolved during the one temperature variation, and some of them evolved low temperature. Yes. Is it okay?
PPT presentation screen টা একটু বড় করুন.
Okay, just
presentation screen টা.
Okay. Is it okay?
No, it is not visible.
Is it okay? Hello?
No, sir.
What happened? Just a minute. What happened here?
অরিন্দম পোদ্দার, আপনি আপনার স্ক্রিনটা শেয়ার অফ করুন।
ঠিক আছে? Is it okay?
No sir, actually this
very small.
অরিন্দম পোদ্দার, আপনি আপনার স্ক্রিনটা শেয়ার অফ করুন।
Just okay. I thought probably this is due to that one. Okay, there are some technical problems obviously there. Is it okay? I think is it? Can you see these slides?
No, sir.
You can't see.
We can only hear you.
Okay,
The
so I
screen
think
is not
sharing, let me share again. Okay, I thought.
Okay.
On your resume your presentations. Why it is called? Anybody can help me?
স্যার স্টপ প্রেজেন্টেশন করে আবার নতুন করে প্রেজেন্টেশন শুরু করুন।
এটা তো আসছে না, resume stop screening sharing.
ধরুন দিয়ে আবার নতুন করে শুরু।
হ্যাঁ, কিন্তু কোথায়? আচ্ছা, okay, I got it. Your entire screen here. এবার এসছে?
No.
No, sir.
Oh,
Okay, sir.
it is visible now, sir.
Okay, sir.
হ্যাঁ আসছে।
আসছে?
ফুল স্ক্রিন করুন। হ্যাঁ স্যার।
Okay, sir.
এসছে?
No.
ওকে স্যার।
Okay, sir.
Okay. Okay, so now come to that point. So now I told you the relations between that COVID virus with the climate changes. Now come to that how COVID-19 spreads. I am almost close to this couple of slides, almost close to my talk. So there is a much to learn about newly emerged COVID-19. All of you know, based on what is currently known, COVID-19 and SARS spreads is thought to occur mostly from person to person. Obviously, it's a human-to-human interaction. Now, bat cannot infect. The bat, even right now, that any bat or pangolin virus cannot infect. Whatever the changes is done. But right now, only spreads can be done human via respiratory droplets. All of you know the droplets. So when if you cough, when if you talk, when if you the breathes, so there are some the whatever the droplets, whatever the water molecules comes from your mouth, and that water molecules contains the virus, and that virus obviously, if any close peoples, they take your droplets and they infected, and ultimately the infection spread. That's the way can spread. Being within approximately 6 feet, 2 meters of a patient COVID-19 for a prolonged period of time from a patient with the COVID-19. Infectious secretions may include sputum, not only the droplets, sputum, serum, blood, and respiratory droplets. Even the urine and the the stools also. If close contacts occurs while not wearing all recommended PPE, all of you know this healthcare people, they take their and for the common people, I will come to that WHO, some of the resolutions, mostly the mask is enough. So now the risk factor of the COVID-19. So this is the CDC, CDC's data. CDC's data is early persons above the age 50, underlying disease like diabetes, Parkinson's disease, cardiovascular disease, this is called the comorbidity, whatever, the comorbidity, very popular. And demographically, it was that time says that one states of the China. Right now, I'm telling whatever the whole world, wherever this, the epidemic happened. So if anyone comes from this region, they are, they can spread this virus to other one. Healthcare workers carrying of patients with the COVID-19, they are very much. And then based upon the data collected from the China, the greater than 10 years, no death, between 10 to 39 years, fatality 0.2, between 60 to 69, fatality 3.6, and between 70 to 79, 8%, and greater than, and then 80 years is the most infectious. So that is the risk. Everybody is known. And right now, I am telling you very recent observations. It came out in the Nature Medicine paper. So what is that one? So this is very important, So now, if you see that many peoples are right now, home quarantine, or if they go to the hospitals within a 4 or 5 days or 14 days, they recovered and come back to the home. But certain peoples are going for that ventilations, or they need a support, health support, the respiratory support, or some of them have died. So what is the fact between? Because I told you this virus, whatever the genes they have, they do not carry any death-like genes. They are like a normal flu viruses. But why it has happened? So now, one group in the what they did, they took the genome, they took the virus, collected the virus, almost 8 to 9 lakhs patients, and they they divided these two patients in two cohort. One cohort, so the patients, those who are already in the ventilation and they died, and another cohort, those who are normally recovered. So these two cohort, so they took the virus from these patients, almost 9 lakhs. This is a huge number, and then sequenced, and we call that genome-wide analysis. Right now we can sequence the whole human genome. They sequence the whole RNA and then whole genome, and then analyzed. So not RNA, actually the human genome, human DNA, and then analyzes what is the variation between this cohort and that cohort. Those who are going to the ventilations and they dying, another one, they're normally recovered. So they came into the conclusion is very good observations. So this is a chromosome 3. They have found there is a cluster of genes. There is a 6 genes cluster of genes. They have a variation compared to those who are normal. So this is called what the kind of variations? I told you what is variations in the when I analysis that RNA of the virus genome, but this is the human genome. So now what is we call the single nucleotide polymorphism. This is all the gene. So these are the genes, single nucleotide polymorphism variation. What is that one? So all of you know that 3 nitrogen base out of 4 nitrogen, 3 nitrogen base make a 1 amino acid. So now think about ATG. ATG is a methionine. So some people encodes ATG as a methionine, and some people AGG also encodes as a methionine. So this is called the single nucleotide polymorphism. They encode the same amino acids, but their amino acid, single amino acids changes. We call the single nucleotide polymorphism. So these groups, they have found in the chromosome 3, this almost 6 genes, they are single nucleotide polymorphisms compared to the normal. And all these genes are nothing but the inflammatory genes. So they said, hopefully, these groups belongs to this single nucleotide polymorphism. As a result of the infection, they are basically all these respiratory systems, become elevated in their body. They have made another very good observation. They made infections with the blood group, ABO blood group. What is that one? So they said that O group is a O minus, or O group is a less infectious compared to less infectious, Where is that? A group is a morely in the susceptible to the infection. So this is the observations have been made. This is a recent data, almost 1 month before. So not 1 month, before than 1 month. So this is a very recent data, and huge amount of the the analysis genomes. The almost 9 lakhs patients they worked. So obviously it has a meaningful. So now come to that. Right now, this is one of the very important, and then peoples are very much very much they're but curious about the therapy of the virus, whether the vaccine, whether the drugs or something like that. But before that, telling you, virus doesn't work with the antibiotics. Antibiotics basically the the bacterial infection. Virus doesn't have. But what the scientists did, basically the clinical scientists, when this is we do not know, still we do not know what drugs are effective for these viruses, how it prevent the viruses. So now actually what the clinical scientists they did, so whatever the whatever the drugs actually the available in the market, or actually available in the market, they used different patients, and then suppose they used on the 200 patients some of the specific drugs. So as a curiosity they did, and then they got some good result. So on the basis of that one, some of the drugs are found effective, but still, so the data suggests there is no drugs have been approved for the specific for this this COVID-2 infection. But some kind of data, I'm telling you, remdesivir, lopinavir. So these are the data are right now came to the phase 3, phase 4. So these are the drugs. These are the non-new drugs. This is drug means these drugs are already present in the market. So scientists or the clinical scientists, doctors, they found these compounds are effective in some of the patients, and as a result of some of the phase trials have been done, and they have now some of them phase 3, phase 4. Phase 4 will be very close, and some of them phase 1, phase 2. So this is the way scientists are trying to develop the drugs for these infections. Okay, now and then monoclonal antibodies. The monoclonal antibodies, that is a antibodies treatment is also available in the market. Antibody treatment for the cancer, antibody also treatment used for the arthritis. So scientists also found some of the antibodies, monoclonal antibodies, are also useful for these virus infections, and then they have started that phase trials, and ultimately some of them are phase 3, early phase 3, phase 2. So hopefully some of the drugs, some of the antibodies may come into the market very soon. So now there are another very popular one, that is a immune modulating natural products. All of you know that. So people are consuming lots of vitamins, lots of the different different types of the non-natural products like a garlic or something like that for this one. But I tried to find out, is there any meaningful or not. I found some of the I gave you the reference, some of the journals, this natural product they use for the evaluated some of the other viruses, not the COVID viruses. Thank you. So as a see the garlic evaluating the herpes, the vaccinia, parainfluenza, fresh ginger, human respiratory syncytial virus, cinnamon used for the H7N3 influenza virus, tulsi H9N2 virus, ginger SARS-CoV virus, earlier SARS virus, and then black tea also used for the earlier SARS-CoV virus, and then vitamin D, zinc elsewhere. And these are all the references. But again, I'm telling you, these are the natural products. It's not the drugs. It's not the drugs. Basically, people are saying immunomodulating, but I don't know what immunomodulating means, because immunomodulating means whatever the immune genes we have, either they are regulated by this product, we don't know, because the gene, all these immune systems developing the cytokines, whatever we have genes, the genes are expressed. But now the question is whether these natural products can regulate our or can express our genes. We don't know all these things, but whatever I found in these references, they used this product, these natural products are used or evaluated on this kind of virus, and they got some fruitful result. So that much I can say. So now drugs using the SARS-CoV and MERS-CoV. So there are some, already some of the, SARS-CoV, earlier SARS-CoV and MERS-CoV virus have some of the drugs also used earlier, and that virus, that drugs are also right now using for this COVID-2 viruses. But now coming to that vaccine, because ultimately vaccine is the vaccine is the real solve of this infection. So now, even whatever the proposed, whatever the idea of the scientist we have for the treatment, first we have to do the experiments, and we have to confirm the experiment. That's called the proof of concept. So when the proof of concept is very clear, then it goes to the pharmaceutical industry for developing that one. So now how this vaccine proof of concept was developed. This is a Cell paper. So one of the group, all of you know, that is the Oxford groups in the UK. What they did, they took the virus. They took the two types of virus. This is a chimpanzee. This is a chimpanzee COVID virus, and this is a human COVID virus. And they have found chimpanzee COVID virus. This is a spike protein. They can easily interact with our AC2 protein and can enter the human cell. This is a chimpanzee, the COVID virus with the same AC2 protein. So with that idea, they took this protein and made an antibody, and then they injected into the human cell, and then now they charged with the human COVID virus, and they found it can protect. It cannot enter into the AC2. This is called the proof of concept. So now with that proof of concept, the vaccines are now developing in the market. So at least, so now whatever the data, this is a little bit earlier data, actually 1 month before. So whatever the data vaccine, I tried to understand, I tried to know where is the status of these vaccines. So there are almost, I found 80 types of vaccines have been developing in that all over the world, and including this vaccine is the, whatever I told you, there's a Oxford virus, Oxford groups. So these vaccines are right now phase 1, phase 2, and then this is the Oxford vaccine. Almost they completed the phase 2. They are in the early phase 3. But so you have to go phase 1, phase 2, and phase 3. Ultimately, then you can come to that the human trial. Then come to the market. So hopefully, if all this phase 3 trial goes well, then we can expect at least these vaccines in the market. But when it is, I don't know. Only I can tell you from my collected data, what is the status of this vaccine right now in the laboratories. So now, almost couple of the last slides. So I told you in my earlier talks. So my objective of this talk to understand what is a myth and what is reality about the COVID-19. So I got it some of the CDC Atlanta and then the WHO report, and from this report I'm telling you what is the myth and what is the fact. So suppose one is the questions: are hands dryer effective in killing the virus? Answer: no. Can ultraviolet disinfection lamp kill the virus? No. How effective are thermal scanner in detecting people infected with the virus? You know that everybody is scanning. So thermal scanners are effective in detecting fever, not the virus. So can spraying all over the body kill the virus? No. Is it safe to receive a letter or a package from China? Yes, it is safe. It is safe. People receiving packages from the China are not at risk for contracting the virus. The question is, there are lots of rumors in the arcade. So I saw the people are also washing the rupees or the note in the home. They are washing with the detergent. But if you see, the virus is very labile. When we are growing the virus in our laboratory, I told you it's basically the outside protein, and then we maintain the specific temperature. If it is altered, we cannot grow these viruses. So think about how long, if virus exists in the other than the host in the in the surface in the anesthesia. Basically the virus, whatever from the droplets comes to the any surface, it will be hardly maximum couple of minutes. Or if you think about very cautiously, 1 hour is enough from the room temperature. So after that one, it should not be. It should not be. There is no, there is no what is called the research. The data I found, this virus can exist in the surface for a long time, 24, 48 hours. Basically, logically, it should not. So now, can pets at home spread the virus? No. So there are also rumors that some of the pets in the New York, the tiger was infected. It is not. Do vaccine against pneumonia protect against the novel coronavirus? We have some pneumonia vaccine, but can it protect because it is an infection associated with the pneumonia? No. Can regularly rinsing your nose with the saline help prevent infection with the virus? No. Can gargling mouthwash protect from infection from the virus? No. It helps to that giving you the comfort, but it cannot protect you from the infection. Can eating garlic help prevent infection with the virus? No. Does putting on sesame oil block the new coronavirus from entering the body? No. Does the new coronavirus affect older people or are younger people? People of all ages can be infected. Are antibiotics effective? No. Are there any specific medicines to treat the virus? No, I told you. So couple of drugs are in the trial, but still there is no such drugs available in the market which can protect the virus. So now all these informations I gathered, I tried to give you a one brief idea about the protocol of this virus treatment, because still FDA, Federal Drug Administration, they did not come out in any specific protocol, but whatever the informations all over the world I collected, and I give you a brief protocol. What is that one? Preventive strategies. Obviously, you have to take the preventive strategies. So maintain social distance, wearing mask, and using hand sanitizer. Vaccines, obviously, vaccines are coming out. Immune boosters. Immune boosters, obviously, vitamin C, and then some of the people, I told you, some natural products, immune boost, but how does it? I don't know. But viral anti-inhibitors. So some of the viral anti-inhibitors, you can disinfect your area or inhibit the virus infection. Management of odds and cytokine storms. So basically, this virus, what they do, they basically express our immune system, overexpression of our cytokine system. As a result of the breathing problem, there is a the mucus developed in our lung, and that's the reason why they are running out of, they are going for the respiratory problems. So management of these cytokine storms. Monoclonal antibodies, I told you monoclonal antibodies, some of them are split and phase 1, phase 2. Hydroxychloroquine. There are a couple of the papers have been found. There is some in favor of the hydroxychloroquine, some of the in favor against of the hydroxychloroquine. But still, some of them said hydroxychloroquine or may work, but still it is not the standard protocol. Supportive therapies like antibacterial, corticosteroid, natural products, and therapeutic option, repurposing antiviral drugs. Whatever antivirus drugs I showed you, it is also in the trial. Novel antivirus drugs, some of the novel antivirus drugs people are already developing, but still it's in the trial. Okay, anti-malarial, anti-helminthic, anti-parasitic drugs, people have used some of the cases and they found some of them are positive. And then interferons inhibitors, natural killer cell cytokine T cells, vaccines, mesenchymal stem cells. I found lots of stem cells therapy, but still are in the clinical trial, or some of them is the research, the experiments. Even last one is the ACE inhibitor, that means the receptor angiotensin converting enzyme inhibitor. There are some the inhibitors are available, those who control, those who are given for the pressure control. So maybe some of them have reported they also used for the effective for this inhibitions of the virus. But still again, I'm telling you, FDA did not come, or our ICMR, Indian Council, they still did not come out in the any standard protocol all over the world. So this is the this is the information I collected from the different publications. And now the last slides, that is the take-home message. I think this is the most important. So that is the follow the rules. What is that one? Reduce your risk of coronavirus infection. Why? How? Clean hands with the soap and water or alcohol-based hands rub. Basically, any detergent, if you use what the detergent does. Detergent basically denaturate the protein. Whatever the virus outside protein, if it is denatured, virus lost its infection ability. So use any kind of detergent or any kind of soap you have in your home. Cover nose and mouth when coughing and sneezing with the tissue or flexed elbow. Actually, we have to develop this habit. Cover nose and mouth when coughing. Avoid close contact with anyone with cold or flu-like symptoms. Thoroughly cook meat and eggs. Thoroughly avoid unprotected contact with the live, wild or farm animals. So as actually there are some incidents already happened. People already know some of the virus evolved from the meat market. So that's why the WHO report, avoid unprotected contact with the live, wild or farm animals. So now come to that I told to follow the rules. So what is that once? So use mask when outside. I think right now all on a sudden this virus will not stop. So obviously the infections will be coming out, but it will not be zero. So hopefully it will be remain for a couple of months or maybe couple of years in this earth. So obviously we have to develop our idea to use mask when you go to the outside. Mask is enough to prevent the droplets, the infection through the droplets. Wash hands before touching nose, eyes and mouth. So this is the habit you have to develop. Wash hands before touching. And basically the children, we have to we have to train the children with, they should not touch their nose, eyes and mouth or eating. They should wash the hands. And last, this is the most important, stop spitting in the public place. So just I like to, personally I'm just requesting all over that, mostly the students, you please take care. You please take care. You should not spit in the, because we have a very bad habit in India. We spit whenever, whatever in the public place. So people are chewing the chewing gum or chewing the pan, whatever the all the betel nuts, and then whatever the pan product, they're spitting on the public place. So that's not only the virus infection. Think about the India. Every year 8 to 9 lakhs people are infecting by the tuberculosis. So tuberculosis, and 4 lakh people dying every year by the tuberculosis. And the tuberculosis is also spreading through the spitting, and not only tuberculosis, many infectious diseases through the spitting. So my dear students, just to think about, stop this habit. It's not only you, when you are in the public place, if you see someone, just don't let them spit on the public place. Raise your voice against the public spitting. And our world, this is our world. Virus will be evolved naturally, and virus will be change their host. But we have to take care of our this world. So this is our responsibility. So with that request, so again I'm requesting you. So just follow these rules. Use the mask, wash your hands, and stop spitting. Even not only you, don't spit other people. Don't let the other people spit on the road. At least raise your voice. And with that, with that attitude, change our attitude, we can keep our our neighbor clean and stop this infectious disease. So with that one, this is the end of my talks, and thank you again. And if you have any questions, please let me. I will try to answer my answer. Okay, thank you.
Thank you, sir, for such an enlightening talk. It's very interesting and informative. It's very interesting and informative.
Thank you. So now question and answer will be at the end of the sessions, or right now I can?
No, sir. Due to the crisis of time, we have to omit the question answer session right now. If time permit, we will continue it after the second session.
Okay.
Thank you. Thank you, sir, for your wonderful talk, sir. Thank you.
Should I stop, right? Okay.
Yes, sir. Yes, sir. Definitely. Thank you, sir, for your great talk. Now, without spending the time, it's time for our second talk. I would like to introduce our second speaker of today's session, Dr. Sayak Roy. The topic that we will present today is COVID-19 diagnosis and prevention. Presently, he is a consultant physician and he is associated with Medica Super Specialty Hospital, Kolkata. He is also a senior class assistant in biochemistry and microbiology. He pursued his MBBS from Bardhaman Medical College and Hospital and pursued his PG DHSC Diabetology from Animalia University. Completed his MRCP from Royal College of Physicians of Ireland. Postgraduate diploma in endocrinology from University of South Wales, UK. He has many renowned publications in reputed national and international journals. He has been invited as speaker in various state and national level conferences. He has presented posters in international level in Berlin in 2019 and in Spain in 2020, presented at ATPD, an international conference of the world diabetologist and endocrinology doctor. I cordially invite you, sir, to please deliver your talk. Thank you, sir. Over to you, sir.
Thank you very much. Am I audible first of all?
Yes, sir, you are.
The first thing is that I cannot see my own slides. That's a basic problem. Can you people see that?
No, sir.
Oh, that's great. That creates the problem.
You can just put the slide on presentation mode.
Yes, I've done that, but I do not know. You're presenting your screen, showing you're presenting your screen now. No screen is available. So, what to do now?
The slides are visible. Just put the slide on presentation mode.
The slides are visible?
Yes,
Really?
yes,
Yes.
So,
What is visible? Just put the slide on presentation mode.
it's okay?
Yes, sir, it's visible.
Just I will take just few seconds, okay? Just tell me whether it's moving or not. It's moving?
It's moving. It's moving.
Okay, great. So straight away, I'm going to the diagnosis and the prevention with after the fantastic presentation by my previous speaker. So I'll be delivering my speech both in English as well as in Bengali because অনেকেই হয়তো মানে পুরো ব্যাপারটা ইংলিশ নিলে হয়তো অনেকের প্রবলেম হতে পারে. So it's like I will combine both of the languages. I will try to do my best, okay? And can all the people, other people, just please unmute yourself? So COVID-19 diagnosis. We know after all the elaborate presentation by my previous speaker, I'm definitely not going to get into details about how it is infecting and how it is doing the damage to our system. The thing is that it is over here. It is here to stay till the vaccine is out. But the thing should be very clear in our mind whether what is the present scenario and what is the scenario in India. If we go into details, the WHO will tell you that the case scenario, the case fatality rate is almost 3.75%. So it's not a huge thing. কিন্তু নেই যেটা সবথেকে এখানে আমি দেখছি ম্যাক্সিমাম জায়গাতে প্রচন্ড ভয় পাইয়ে দেওয়া হচ্ছে যে হলেই অ্যাজ ইফ যেন হলেই মারা যাচ্ছে, তাই জন্য পেশেন্ট একটা সোশ্যাল স্টিগমা তৈরি হয়েছে, পেশেন্টের কাছে যেতে চাইছে না। অবভিয়াসলি যাওয়ার দরকার নেই, কিন্তু দূর থেকে তো কথা বলতে না, দরজা জানলা বন্ধ করে রেখে দেবো, আমার ওর হয়েছে আমার কেন, আমার হবে না বাবা, দরজা বন্ধ করে দে। It's ridiculous. It doesn't spread like that. In the fear, first of all, come out of the fear and be rational in your approach যে কোথায় কোথায় আমরা প্রিভেনশন করতে পারি, আর হলে কি কি সিমটম আছে, আর হলে আমরা টেস্ট করাবো। সোশ্যাল স্টিগমাতে আমি অনেক পেশেন্ট পাচ্ছি যারা টেস্ট করতে চাইছেন, তাদের একটাই কথা, ডাক্তারবাবু করালে আমাকে তুলে নিয়ে চলে যাবে অ্যাম্বুলেন্স নিয়ে। এটা হচ্ছে না, দিস ইজ নট দ্য সিনারিও। কোথায় রাখবে ভাই, রাখার জায়গা কোথায়? হাসপাতাল পুরো ভর্তি, কোথাও রাখার জায়গা নেই। সুতরাং তোমাকে রাখতে হবে। And the best thing is that go for home quarantine. PDF of Ministry of Health that fill it up and see for yourself. স্পষ্ট লেখা আছে, কোনো কনফিউশনই নেই। না শোনে বা পেশেন্ট যদি অযথা ভয় পায় তাহলে তো আর কিছু বলার নেই। We are there to help you, but please come out of the fear. After saying that, come out of the fear, come out of your masks and transmit the virus to somebody else. So be careful. I am saying to come out of the psychological fear, the psychological barrier, not of the physiological, the social distancing that we are supposed to maintain, the hygienes that we are supposed to maintain. Please do not come out of that. That will be disastrous if you do that. Of course, if you can scenario, it's only 2.05%. কিন্তু আপনি যদি নিউজ চ্যানেল দেখেন, আজকে এত মারা গেল, কালকে এত মারা গেল, আরে বাবা, focus on the number of patients infected and the number of deaths, and then find the percentage of death. Please, for God's sake, do not spread the fear. সত্যি কি খুব বেশি মনে হচ্ছে? আরে হার্ট অ্যাটাক 60 to 80% পেশেন্ট মারা যায়, পেশেন্ট গ্যাস অম্বলের ওষুধ they just don't come up. They say এটা আমার গ্যাসের ব্যথা ডাক্তারবাবু. They do not listen to people. They just keep on listening, and that's why we lose patients having actually a myocardial infarction. That's disastrous. So please be aware of these facts. যখন একটা জিনিস দেখবেন, look into the case fatality. আজকে এত হলো, আজকে 54,000 হবে, স্বাভাবিক, that's normal, because the virus is highly infectious, খুব তাড়াতাড়ি স্প্রেড করছে, বাট দ্যাট ডাজন্ট মিন যে সবাই মারা যাচ্ছে। এবং যারা মারা যাচ্ছেন, maximum to maximum, most of them are having an underlying comorbidity, age being the most common, like our previous speaker also said. And of course, there are many other things, age being the most common. After that, we have lots of comorbidities like hypertension, like diabetes, like renal failure. Excuse me.
একটু ইকো হচ্ছে স্যার, ইকো হচ্ছে স্যার একটু।
ঠিক আছে এখন?
ঠিক আছে,
ওকে ফাইন। সো এবার ইন্ডিয়ান সিনারিওটা যদি দেখি আমরা, আমাদের ব্যাপারটা মাথায় রাখতে হবে যে it's not that people will die, all of the people will die. In fact, I have many patients who have just recovered, just বাড়িতে থেকে তারা রিকভার করেছে। Of course, I have told them what are the danger symptoms. Not me, এটা আমি বলছি না, আমি তৈরি করিনি। It is there in the internet, the Ministry of Health, in the Ministry of Health website, on the Government of India website, Government of West Bengal website, সব জায়গাতে স্পষ্ট লেখা আছে ভাই, এখানে কিচ্ছু কোনো এদিক-ওদিক করার নেই। যা লেখা আছে প্রোটোকল ফলো করো, you will be safe. সুতরাং অযথা ভয় পাওয়ার দরকার নেই, টেস্টটা করান, কারণ আপনার থেকে আর 5টা লোকের হতে পারে, এটা সব থেকে বড় কথা। আপনার হয়তো কিছু হবে, কিন্তু আপনার বাড়িতে যদি বয়স্ক ঠাকুমা দাদুরা থাকে, এখনো ইন্ডিয়ান ফ্যামিলি তো we still we try to stick together, you know, and like not like the other people, not like the Caucasian people, not the Caucasian population where people live separately. যেখানে বিদেশে আমরা দেখেছি যে বাবা মা আলাদা থাকে, ছেলে আলাদা থাকে, দাদু ঠাকুমা আলাদা থাকে। It's not like that. We still believe in staying together. It's very good because you have the manpower with you, but be cautious, কারণ তোমার হয়তো কিছু হবে না, ইয়াং এজে হয়তো তোমার ম্যানিফেস্টেশনটা খুব বেশি আসবে না, সামান্য ম্যানিফেস্টেশন তুমি আর ইগনোর করবে, বাট তোমার জন্য বয়স্ক যা 80 বছর বয়স, তার হয়তো প্রবলেম হয়ে যেতে পারে। সো জাস্ট বি কেয়ারফুল। হলে টেস্ট করো, আইসোলেশনে থাকো যতক্ষণ রিপোর্টটা আসছে। দ্যাটস দ্য ইজিয়েস্ট ওয়ে। সো যদি সিনারিওটা দেখি আমরা, দ্য কেস ফ্যাটালিটি ইজ অনলি 2.05%, and আই এম ডেফিনেট দ্যাট এরপরে আর নিশ্চয়ই লোকজন মাথা খারাপ করবে না যে প্রচন্ড রকম ফেটাল ব্যাপারটা কম, রাদার দ্যান স্প্রেডেবিলিটি অনেক বেশি। And this is just a thing which I will not go into details. Thing is that this is how the virus enters, is from the nature's medicine. তারপরে ডিরেক্ট সাইটোটক্সিক এফেক্ট আছে, ভাইরাসটা ডিরেক্টলি এফেক্ট করতে পারে, ভাইরাসটা ডিরেক্টলি সাইটোকাইন স্টর্মটাকে তৈরি করতে পারে। সাইটোকাইন স্টর্ম ইজ সামথিং লাইক দ্যাট, আমার বডিতে রেসপন্স দরকার, দ্যাটস ফাইন। You get a virus inside you and you develop an antibody response and you develop the normal cytokines, মানে কিছু chemical mediators আছে যেগুলো বেরিয়ে আমাদের ওটাকে নষ্ট করবে, that's fine, up to this it's fine। কিন্তু কিছু পেশেন্টের ক্ষেত্রে কি হয়, ওভার এক্সাজারেটেড রেসপন্সেস আসতে আরম্ভ করে, মানে যেটা দরকার নেই সেখানে হিট করতে আরম্ভ করে। So it damages all the systems. These are the patients who will finally get the severe form of the disease. It's only 7 to 10% of the patients. So not the 90%. The 90% of the patients will be maintaining a mild to moderate course of the disease. And mild to moderate course সব কিছু দেওয়া আছে, আমাকে আলাদা করে এখানে ডিটেইলস বলার কিছু নেই। জ্বর আসছে, কাশি আছে, জ্বরটা 7 থেকে 10 দিন থাকতে পারে, কাশি আসতে পারে, নেবুলাইজ আমাদের ইনহেলার যেমন ইউজ করি, যাদের ব্রঙ্কিয়াল অ্যাস্থমা আছে তারা নেবে, যাদের যেটা নরমাল ওষুধ তাদের সেটা খাবে, তার সাথে অন টপ অফ দ্যাট সিমটোম্যাটিক রিলিফের জন্য যেটা দরকার সেটা খাবে। আর আমাদের যেটা গাইডলাইন যেগুলো দেওয়া আছে যে এই ওষুধগুলো এখন এক্সপেরিমেন্টাল লেভেলে আছে, সো obviously data is very sparse, but of course we have some of the drugs like remdesivir, like favipiravir, which are effective in the mild to moderate. And of course when you give remdesivir in a severe case, it will not help you. So hit it early when the cytokine storm is just beginning. That is the time when you will get the maximum benefit from these kind of drugs. Just to say only few things about this one. So how to detect COVID-19? Of course, আপনি কি কারো কন্ট্যাক্টে এসেছেন, এটা যখন প্রথম শুরু হয়েছিল তখন এটার কাজে লাগতো, এখন কিচ্ছু কাজে লাগে না, কারণ এখন এটা কমিউনিটি স্প্রেড হচ্ছে। সিম্পলি যতই আইসিএমআর বলুক যে কমিউনিটি স্প্রেড হচ্ছে, it's just community spread. We are all getting thousands and thousands every day. I'm getting 6 to 7 patients of COVID যারা কোনো ট্রেসই করতে পারছে না, করতে পারছে, but of course you are getting that, so it's useless asking them right now. Of course, it's helpful to trace the patients. Contact tracing, মানে ওর সাথে যারা আছে ফ্যামিলিতে তাদের যেন না হয়, তাদেরকেও যাতে টেস্ট করে নেওয়া হয়, that's a good option. Clinical symptoms, we all know, I will elaborating that one. A laboratory testing, we know the RT-PCR is there, and antibody detection, we do not do that. Antibody detection, that will just create confusion much more rather than do any help. And X-ray or CT scan. CT scans are very, very, very sensitive. Not at all X-rays. X-rays are not at all recommended as a first line. I would rather go for a CT scan in a patient who is refusing our RT-PCR, and we have found the patients having HRC, there is a high resolution CT scan of thorax showing positive results, and the patients are getting TrueNAT negative. Finally, the patients are getting RT-PCR positive from another lab because that was a rapid test, and finally you do an RT-PCR and you get the positive results. You know, there is a time frame when this test will be positive, when antibody will be positive, so don't jumble them up. একসাথে দুটোকে এক করে দেবেন না, কখন অ্যান্টিবডি করব আর কখন RT-PCR করব, সবকটার একটা টাইম সেগমেন্ট আছে। এবং RT-PCR যখন করছি তখন কোথা থেকে স্যাম্পলটা নিচ্ছি, কিরকম ভাবে নিচ্ছি, কিসে ট্রান্সফার হচ্ছে, সবটার উপর ডিপেন্ড করে যে পজিটিভ আসবে না নেগেটিভ আসবে। যদি পজিটিভ হয়, লো ভাইরাল লোড থাকলে অবভিয়াসলি তাদের কিরকম, আর হাই ভাইরাল লোড থাকলে তাদের কিরকম। দিস ইজ সামথিং হুইচ আই হ্যাভ হাইলাইটেড। ফিভার, কাফ, ঠিক আছে, এগুলো তো সবসময় আপনারা ফোন করলে, একে অপরকে ফোন করতে গেলে এখন শুনতে পাবেন, 5 মিনিট ধরে এটা শুনতেই হয় আমাদেরকে। Shortness of breath, difficulty in breathing, only it occurs in about 20% of the patients. 80% এ কিন্তু আমরা যেটা পাচ্ছি as clinicians, we are getting diarrhea with fever and loss of taste and smell. গন্ধ পাচ্ছি না ডাক্তারবাবু, 2 দিন ধরে গন্ধ পাচ্ছি না, নাকে কোনো গন্ধ নেই, জ্বর হয়েছিল, হ্যাঁ ডাক্তার 2 দিন আগে জ্বর হয়েছিল, মোস্ট লাইকলি কোভিড। এটা খুব প্রচন্ড কমন এখন, ডায়রিয়া, নিউ লস। রিমেম্বার, একজন পেশেন্ট তার নার্ভ ড্যামেজ অলরেডি আগে থেকে ছিল, সে এমনি গন্ধ পাবে না, সুতরাং তাকে জিজ্ঞাসা করার কোনো মানে হয় না। সো লস অফ টেস্ট অ্যান্ড স্মেল অ্যান্ড ডায়রিয়া অ্যান্ড ফিভার, all these things coupled together. কাফ নাও থাকতে পারে, কাশি অনেক ক্ষেত্রে পেশেন্টের হচ্ছে না, বা পেশেন্ট হয়তো বলছে না, ভয়ের চোটে বলছে না যে না আমি ডায়রিয়া বলে ধরা পড়বে না, আমার কাফ বলে ধরা পড়ে যাবে। দ্য থিং ইজ দ্যাট উই নো ইউ ম্যান, সো প্লিজ এগুলো লুকিয়ে কিছু লাভ হয় না, আলটিমেটলি ক্ষতি হয়। সো কাম আপ, tell the doctors what problem you actually have, then only we can tell you whether to go for COVID-19 test or do not need to go for any COVID-19 test. Muscle ache, obviously না। and আরেকটা জিনিস অদ্ভুত ভাবে পেশেন্টদের পাওয়া যায়, আউট অফ প্রপোর্শন উইকনেস, প্রচন্ড উইকনেস, সিভিয়ার উইকনেস থাকছে। এটা আমি বারবার বলছিলাম যে RT-PCR কখন করব, অ্যান্টিবডি টেস্ট কখন করব। যদি খুব ভালো করে স্লাইডটা আমরা দেখি, নেজোফ্যারিঞ্জিয়ালস মানে নাক থেকে, যেটা নাকটাকে তুলে নাকের ভেতরে একটা সোয়াব স্টিক ঢুকিয়ে দেওয়া হয়, অথবা ওরোফ্যারিংস মানে হাঁ করতে বলা হয়, সেখান থেকে টানা হয়। নর্মালি নেজোফ্যারিংস এটা বেটার আসে। তো নেজোফ্যারিংস অথবা ওরোফ্যারিংস অথবা ব্রঙ্কোস্কোপি করে ব্রঙ্কো অ্যালভিওলার লাভ আছে, মানে ব্রঙ্কোস্কোপি মেশিনটাকে ঢোকানো, ব্রঙ্কোস্কোপ মেশিনটাকে ঢুকিয়ে জল দিয়ে ইরিগেট করে সেটাকে টেনে সেখান থেকে আমি আরটিপিসিআর করছি। সো এনিথিং ক্যান বি ডান। দ্য থিং ইজ দ্যাট আমাদের এটা দেখতে হবে যে ফার্স্ট উইকে ম্যাক্সিমাম পজিটিভ। রিমেম্বার দিস ওয়ান, ইনফেকশন হলো ফার্স্ট উইক, ইউ গেট ভাইরাস, আফটার দ্যাট ড্রামাটিক্যালি কমতে থাকবে, কিন্তু আরটিপিসিআর পজিটিভ থাকছে আফটার 3 উইকস, 3 সপ্তাহ বাদে। হ্যাঁ রাখতে পারবো 3 সপ্তাহ আগে জ্বর হয়েছিল, ভাই আরটিপিসিআর কোনো লাভ নেই, কারণ কিছু লাভ পাবো না, আমি পাবো না, জাস্ট সিম্পলি পাবো না। তখন কোনটা পাবো? আইজিএম আইজিজি, কারণ তখন বডি আইজিএম আইজিজি তৈরি করতে শুরু করেছে। এবার ফার্স্টে আমরা যখন ডেঙ্গুতে করছিলাম আইজিএম উইদিন 5 ডেজ আমরা পেয়ে যাচ্ছিলাম, এখানে কিন্তু তা পাচ্ছি না আমরা। এখানে কিন্তু অলমোস্ট লাইক 2 উইকস to 3 উইকস লাগছে পুরো পিকটা উঠতে। সো ডিফারেন্ট ভাইরাস, different results in the IgM IgG varieties. This has to be remembered. কোন টেস্ট কখন করব? আর স্টুলেও পিসিআর আরটিপিসিআর পজিটিভ পাচ্ছি, কিন্তু থ্যাঙ্কফুলি আরটিপিসিআর পজিটিভ পেলেও ভাইরাল কালচার নেগেটিভ এসেছিল। সুতরাং একটা জিনিস যদি আমরা ভাবি যে আমি আজকে যদি সাপোজ আমার এখানে ছুরি মারলো কেউ, আমার হার্টটা বেরিয়ে গেল, আমার হার্টটা তুমি দেখতে পাচ্ছ, ফাইন। আর আমি যখন একটা ইকোকার্ডিওগ্রাফি করছি উপর থেকে দিয়ে, সেটা তো আমি হার্ট দেখতে পাচ্ছি। একটা ক্ষেত্রে আমি মারা গেছি, আমার হার্টটা বেরিয়ে গেছে, সেটাতে আমার হার্টটা দেখা যাচ্ছে। আর একটা ক্ষেত্রে ইকোকার্ডিওগ্রাফি, আমি বেঁচে আছি, আমার হার্টের উপর ইকোকার্ডিওগ্রাফি এটা ওই প্রোবটা ঘোরানো হচ্ছে, আমি হার্টটাকে উপর থেকে দেখছি। দুটো ক্ষেত্রে কিন্তু হার্ট দেখছি। জাস্ট লাইক দ্যাট পিসিআর যদি আমি করি, একটা ডেড ভাইরাস হলো, আমি ডেড ভাইরাসটাকে আমি পিসিআরটা নিলাম, যখন ভাইরাসের আরএনএটা নষ্ট হয়নি সেই অবস্থায় আমি তুললাম, সেই অবস্থায় তুলে আমি আরটিপিসিআর পজিটিভ পাবো, বাট দ্যাট ডাজন্ট মিন দ্যাট দ্য পেশেন্ট ইজ অ্যাকচুয়ালি ইনফেক্টেড। সো দেয়ার ইজ এ বিগ ডায়ালেমা নাওয়াডেজ, যে কাকে আমি ইনফেক্টিভ বলবো কাকে নয়। বেটার টু গো ফর ইউনিভার্সাল প্রিকশন, 14 দিন থাকুক, বেস্ট ওয়ে। That's the best way that has proved. On 39 days, 29 days অবধি আমরা দেখতে পাই RT-PCR positive, but IgM IgG চলে আসে, so we do not know whether the patient is infective or not. In fact, I believe that the patient cannot spread the disease because maybe the CT value, the cycle threshold value of RT-PCR, মানে হচ্ছে কত ফোল্ড দিলে, সাইকেল ফোল্ডটা দিলে আমি অ্যাকচুয়ালি পাচ্ছি ভাইরাসের পিসিআরটা পজিটিভ। সেটা যত লোড কম, যত ভাইরাসের লোড কম, তত বেশি আমাকে সাইকেলটা চালাতে হবে, তবে পজিটিভ পাবো। তার মানে লো ভাইরাস লোড উইথ IgM IgG পজিটিভ, do not worry. And IgG positive, of course, we should not be worrying, and trials are going on this field also. So this is very important. This slide is particularly very important. And this is how it spreads. We all know that the routes of transmission, of course, the most important is via the droplets. Aerosol production only in case of closed spaces, যখন অনেক গ্যাদারিং হচ্ছে, ক্লোজ স্পেসে আছে, যারা আল্ট্রাসাউন্ড করছে বা যারা ভাইরাস ল্যাবে যারা কাজ করছে, তাদের ক্ষেত্রে হয়তো এরোসলটা কিছুটা রোল প্লে করলেও করতে পারে, নট ফর জেনারেল পপুলেশন। ওকে, সো আননেসেসারি ফিয়ার থেকে বেরিয়ে আসুন। সো পেশেন্ট ইনফেক্টিভ, তার থেকে আরেকজনে যাবে। এবার এই ছবিটা থেকে আমি স্পষ্ট বুঝতে পারছি, যদি দুজনকারই নাকে মাস্ক থাকে, নাক থেকে, প্লিজ বি অ্যাওয়্যার অফ দিস ওয়ান। মাস্ক শুড কভার from this to this, not like this. This is not the way. You're covering only your face. Then why should we be taking swab from your nose? Because the virus is there in your nose. Please cover up your nose. It doesn't mean anything unless you cover up your nose from up to the chin. The maximum benefit. And of course, when both of the people are actually using a mask, a person having a SARS-CoV-2 and a normal person who doesn't have SARS-CoV-2, when both of them are using a mask, the risk of transmitting the virus from the infected to the normal person is minimal. And of course, when till you're using the PPE kit for the healthcare workers like us, মোটামুটি 6 মাস হয়ে গেল, I am seeing patients and till now I am not infected. আমি আইজিজি প্রতি মাসে মাসে করাই। আই ডু নট নো ওয়েদার এসিম্পটোম্যাটিক হয়ে কোনদিন ইনফেকশন হয়ে যাবে বুঝতে পারবো না। সো কিপ অন ডুইং দ্যাট। Still now, few days back, like 7 days back, I did my IgG, it came out negative. And maximum ক্ষেত্রে দেখা যাচ্ছে those who are not using the PPE correctly and as guided by the government, as well as the recommending bodies like the CDC, I do not prefer to go by the WHO all the time, because after all these things that they have done, I've lost most of my faith on WHO. So CDC is a good one till date. So whatever protocols they have laid down, if you follow them, well, of course you can prevent that. You can prevent the infection. You can prevent yourself from getting infected. So direct contact এবং লিফটে উঠছে যখন, তখন থেকে ওই হাত দিয়েছিল ফোমাইট ট্রান্সমিশন যেটাকে বলি, to some extent yes there is chance, যদি খুব তাড়াতাড়ি হয় তো। So 6 feet distance and mask, these are the most important things that you have to remember. Now, infection capability of SARS-CoV-1 and SARS-CoV-2. This is a beautiful journal which I found. যে স্টেনলেস স্টিলে কতক্ষণ থাকে, কপারে কতক্ষণ থাকে, কার্ডবোর্ডে কতক্ষণ থাকে, এরোসলে কতক্ষণ থাকতে পারে, প্রোভাইডেড দ্যাট এরোসল আপনাকে কিন্তু থাকতে হবে যেটা ক্লোজ স্পেসের মধ্যে, আর প্লাস্টিকে 72 ঘন্টা অবধি, স্টেনলেস স্টিলে 48 ঘন্টা অবধি। অফকোর্স আই এম নট দ্য স্পেশালিস্ট ইন দিস কাইন্ড অফ ফিল্ডস, লাইক this is from a very good journal. I have taken it down, and for all the people who are out there, and for all the molecular biologists out there, they can of course go through this journal, and it's a very good one. And for me, what I do is that I keep the package. Of course, I do take up packages from Amazon, from everywhere, so I am not worried about that, because they maintain the protocol. And what I do is that I keep it for 48 hours. That is my take. Okay, I'm not going to tell you that I'm wrong or I'm right or whatever. I'm just trying to follow this kind of protocols, whatever is laid down in this journal. So, how to prevent surface to person transmission? Of course, surface to person, মানে আমি আমার থেকে আরেকটা মানুষের, মানে আমি আরেকটা মানুষ ধরে গেছিল সেটাকে নিয়ে না, কিরকম ভাবে হ্যান্ড ওয়াশ। যদি সারফেস থেকে আসতোই না, তাহলে আমার কাছে সারফেস হাইজিন মেনটেন করার কথাটাই উঠতো না, কথাটাই জাস্ট আসতো না, যদি আমার সারফেস যদি এক্সিস্টেন্স না থাকতো ভাইরাসটা। সুতরাং থাকে, বাট কতক্ষণ থাকে স্টিল ডিবেটেবল, লটস অফ কন্ট্রোভার্সি। ইনফেক্টিভ ডোজ 50 কতটা, মানে কতটা ভাইরাস থাকলে সেটা অ্যাকচুয়ালি ট্রান্সমিট হবে, সেগুলো কিচ্ছু জানি না। সো টু বি অন দ্য সেফ সাইড,
Sir, excuse me, sir. You are not audible. Sir, you are not audible.
তারপরে অডিওটা মিউট হয়ে গেছে।
বৈশাখী সায়ক স্যারকে মিউট করে ফেলেছেন।
শোনা যাচ্ছে?
It's audible now.
এবার শোনা যাচ্ছে।
হ্যাঁ, now okay. শোনা যাচ্ছে।
ঠিক আছে। So again I'm coming back. যখনই whenever I'm not audible, please tell me, okay?
Okay, sir.
Okay. So wash your hands, হাতটা ভালো করে ধোবেন। একটা কোভিড যতই খারাপ, যাই খারাপ করে থাকুক, একটা জিনিস আমি দেখতে পেয়েছি পেশেন্টদের মধ্যে, সচেতনতা টুয়ার্ডস হাইজিন একটু হলেও বেড়েছে। 1%। পেশেন্ট সামনে আমার মুখের উপর কেসে দিত, এখন সেটা অ্যাটলিস্ট বন্ধ করেছে ওরা। থ্যাংক গড। মানে এইটা অ্যাটলিস্ট, এই বলে না সবকিছুর মধ্যে একটা পজিটিভ দিক থাকে, পজিটিভ দিক হচ্ছে আমরা জানতে পেরেছি যে হাইজিন জিনিসটা মেনটেন করা উচিত, যেটা এত বছর ধরে বলেও করতে পারিনি। আর একটা জিনিস হচ্ছে যে আমরা একে অপরের পাশে থাকতে পারি, এবং কিরম ভাবে থাকতে পারি সেটা এই প্যান্ডেমিকটা বুঝিয়ে দিয়েছি, এবং আমাদের কোথায় কোথায় ফ্যালাসি, মানে কোথায় কোথায় আমাদের শর্টেজ আছে। হেলথ কেয়ার সিস্টেমের বাজেট যদি আপনি দেখেন, ইন্ডিয়ার অনলি 3.1% up to 3.4%. What is this? 17% in US. I'm not expecting a 17% rise in your budget for our health, but at least give raffle on. I'm not outside, but please invest করুন, ল্যাবরেটরি তৈরি করুন। এই একটা ল্যাবরেটরি পুনেতে, একটা ল্যাবরেটরি, সেন্ট্রাল ল্যাবরেটরি কেন? সবকটা স্টেটে, সবকটা মেট্রোপলিটন সিটিতে কেন থাকবে না ল্যাবরেটরি? সবকটাতে বায়োলজি ইনস্টিটিউট কেন থাকবে না? আমরা কেন রিসার্চ ওয়ার্ক করতে পারবো না? রিসার্চ ওয়ার্কে এত কম টাকা, আরে করবে কি একটা, যদি আপনি যদি ফান্ডিং না দেন সে করতে কেন, তাকে পেটও চালাতে হবে, অফকোর্স সেই স্টুডেন্টটা সে তাকে পেটও চালাতে হবে। সুতরাং দিস ইজ ভেরি রেশনাল, ইউ হ্যাভ টু ইনক্রিজ ইউর বাজেট on healthcare system. That is very, very important. Pandemic has exposed our skeleton of the healthcare system very vividly, it very much. So do not touch with your bare hands. Once you are outside, please come back, wash your hands, sanitize your hands, then you can touch. Do not touch that before that one, okay? So remember the shed: sleep, hydration, exercise, balanced diet. Balanced diet কেন বলছি? These are lots of things. I'm not going into details. Vitamin C, vitamin D, daily recommended allowance. If you want, I can send you all those stuffs, all the PPT which I have, and you can go through this. I'm not a nutritionist, so I'm not going into details of this. Just the thing, normal খাবারের মধ্যে আমাদের সব জিনিসগুলো থাকে, আলাদা করে মাল্টিভিটামিন খেয়ে ভগবান জানে না যে কি কতটা কি কাজ হয়। এবং সবথেকে মজার কথা, whenever we are seeing a trial, খবরে খুব সুন্দর দেখতে পাই আমি যে এটা খেলে এটা চলে আসছে, এটা খেলে প্রিভেন্টিভ। What is the rationality of the trial? Is it a randomized but blind, double-blinded controlled trial? নাকি একটা আমি প্রসপেক্টিভ ট্রায়াল করে দিলাম? ট্রায়াল ডিজাইন ইজ ভেরি ইম্পর্টেন্ট। সুতরাং ওসব ছাড়ুন, জাস্ট থিঙ্ক অ্যাবাউট দিস ওয়ান: মিট, সেলফিশ, চিকেন, লেগুম, ক্যাশিউ নাটস, এগুলো সবকটার মধ্যে জিঙ্ক যথেষ্ট আছে। জিঙ্ক উইল হেল্প আপ বুস্টিং ইমিউনিটি। ভিটামিন সি কারি জিঙ্কোফিট পাওয়া যাচ্ছে না, জাস্ট পাওয়া যাচ্ছে না, ভগবান জানে কেন ভাই। যদি ভিটামিন খেয়েই যদি করোনা না হতো, তাহলে আমরা ওষুধ না বার করে, ভ্যাকসিন না বার করে ভিটামিনটাকে গুঁড়ো করে দিয়ে দিতাম, হয়ে যেত, করোনা হতো না। কারণ তাহলে লাখে লাখে পেশেন্টে করোনা হতো না। আমাদের ইন্ডিয়াতে তো ভিটামিন খাওয়ার একটা প্রচলন আছেই আছে, কিছু খাবো না, একটা ভিটামিন প্লিজ দিন ডাক্তার। আরে ইউ ডু নট নিড আ ভিটামিন ম্যান। না না, আমার না শরীরটা কেমন দুর্বল লাগছে, খান খান, কি বলবো। So this is where you have to use your rational approach. We have vitamins and minerals in our daily foods. Please use your foods rationally. You can get all the protection from your natural foods and fruits and vegetables. Do not need to buy like vitamin C, vitamin D, all those stuffs from the companies. You do not need to buy that. You can just have a healthy breakfast, healthy diet, healthy food. You will get all the things that you need. But of course, remember, don't get a very feeling like I am taking the vitamin, so I will not be infected. It is not happening, my dear. Lakhs and lakhs of people will not have been affected because most of the people are still on the vitamin. Vitamin D, now this is very important. This is salmon মাছ আমাদের এখানে পাওয়া যাবে না। ভিটামিন ডি টা একমাত্র আমি বলবো যে of course you can take that from a good company, vitamin D, because that is something which we have found in the Indian farmers also. সান এক্সপোজার কিন্তু ফার্মারদের হচ্ছে, যথেষ্ট হয়, স্টিল দেখা যায় ফার্মারদের ক্ষেত্রে ভিটামিন ডি লেভেল ইজ ভেরি ভেরি লো। সুতরাং ভিটামিন ডি সাপ্লিমেন্টেশন can be taken and it's a very safe molecule, so you do not need to worry about vitamin D intoxication unless and until you are like way above, like 50 times above the limited allowed allowance. So you can take it from all these stuffs, or else you can just go for a vitamin D 60,000 which you have from any good company. Again, I'm saying good company. Now talking about the vaccine race, these are the vaccines. আমরা জানি এত কিছুর মধ্যে গিয়ে লাভ নেই। ফেজ 3, ফেজ 2, ফেজ 3, ম্যাক্সিমাম ফেজ 3 তে চলে এসেছে, মডার্না ফেজ 3 তে আছে, হয়তো কোভ্যাক, আমাদের করোনা ভ্যাকটাও আছে, আমাদের ভারত বায়োটেকটাও আছে। সো প্রবাবলি লাইক এন্ড অফ নভেম্বর অর আমি জানি না 15 আগস্ট কি কেন বলে দিলাম আমি বলতে পারবো না, whether it's a political gimmick or something like that, I am not sure. Only what I can tell you is that you have to wait final outcome of these trials, then only you can say whether yes, this vaccine can be given. Of course, the Oxford vaccine tao ashar kotha, so we are all waiting for this vaccine. ele ki shobai peye jabe? Of course noy, karon desh সে তার মধ্যে যদি 3 কোটিও ভ্যাকসিন আসে 133 কোটিতে পেতে পারে না। তাই জন্য সেখানে আমরা কি করতে পারি, ট্রায়াজ মানে আমরা দেখবো যে কোন গ্রুপটা বেশি ভালনারেবল, কোন গ্রুপটা অফকোর্স 65 above they are vulnerable, 70 above they are very vulnerable, they will be given the first lot, of course with them the healthcare workers যারা directly patient deal করছে। তারপর the rest of the general people, বাচ্চাদেরকে তো এখন ধরাই হয়নি ট্রায়ালের মধ্যে, কারণ হচ্ছে we are not seeing, thankfully we are not seeing COVID-19 in that huge amount in case of children, but of course they are getting affected, but they are doing very well. Fantastic news of this horrible disease. I'm not saying deadly disease, it's a horrible disease because they have actually made our life a mess. So it's good that the babies, the children are not suffering to that extent as the Caucasian people. The Asian peoples, the Asian children are not suffering that much. So now I'll be just adding a few myth busters before completing my slides. Can shoes spread the coronavirus? Is very, very low. Okay, so do not have to worry about that. You can just keep them if it will help them prevent with dirt and any waste that comes. So consider leaving them at the entrance of your home, মানে বাড়ি, মানে যখন ঢুকলেন দরজার বাইরে জুতোটা পরে দয়া করে ভেতরে ঢুকে আসবেন না, এটা অনেকের হ্যাবিট থাকে, জুতোটা পরে বাড়ির মধ্যেই চলে যায়, সেটা করবেন না, সেটা এমনিতেও বাজে। আর হচ্ছে মাস্কের ক্ষেত্রে আমরা কি করব না, নন মেডিকেল ফ্যাব্রিক মাস্ক যেটা আমরা করি বা করি না, যেটা করা উচিত নয়, নাকের তলায় পড়বেন না, মানে হয় না। 1 মিটারের মধ্যে যখন আসেন তখন দয়া করে ওটা স্মার্টলি, হ্যাঁ আমার কিছু হবে না ভাই, খুলে ফেললাম, তোর হবে না, তোর থেকে আরো 5টা বয়স্ক লোকের হয়ে যাবে, নাহলে কথা বলিস না। দ্যাট ইজ ডিফিকাল্ট টু ব্রিথ, শ্বাস নিতে পারছি না, আবার না বেরিয়ে শ্বাস নিতে পারছি ভালো না, তাহলে লাভটা কি হলো মাস্কটা পরার? মাস্ক যদি বারবার সামনে যদি হাত দিয়ে নামিয়ে দিতে, এটা তো কোনো লাভই হচ্ছে না, ইটস জাস্ট ইউজলেস। এন্ড প্লিজ ডু নট টাচ দ্য ফ্রন্ট অফ ইউর মাস্ক when you are putting it, put it like this, and when you're removing it, use your hands like this. Am I visible, by the way? Am I visible? Video তে দেখা যাচ্ছে কি আমি যেটা দেখাচ্ছি?
Yes, you are visible.
Yes.
ঠিক আছে,
Yes, you are visible.
এটাকে পেছনে নিয়ে এটাকে এরকম ভাবে খুলবো, আমি সামনে থেকে ধরে খুলে দিচ্ছি, লাভ নেই কিছু, কারণ ভাইরাসটা এখানেই আটকে আছে, আমি ওখানেই হাত দিচ্ছি, কি লাভ হলো? বেকার মাস্কটা পরার কোনো মানেই হলো না। সুতরাং এইগুলো খুব ভেবে চিনতে, এখান থেকে এতটা যেন পুরোটা যেন কভার করে, চীন অব্দি যেন পুরোটা কভার করে, এতটা কভার করলো, এইটুকুনি মাস্ক আমি দেখছি আজকাল বিভিন্ন রকম আমাদের লাভ ইউ ইন্ডিয়া, তারপরে ইস্ট বেঙ্গল মোহনবাগান সবাইকার ছবি লাগানো মাস্ক, এখান থেকে এইটুকুনি, কি মানে ভগবান জানে, আর সেই জানে যে তৈরি করছে, আর যে পড়ে সেই জানে ভাই। সুতরাং প্লিজ একটু ভেবে চিন্তে মাস্কটা পড়ুন, মাস্ক পড়ুন এবং অবশ্যই পড়ুন, কিন্তু ভেবে চিন্তে পড়ুন। বারবার নাক ধুলে গরম জল দিয়ে করোনা চলে যাবে, আপাতত কোনো এভিডেন্স নেই। And thankfully my previous speaker has also said all these things in a very elaborative manner, so I'm very thankful to him. And exposing yourself to the sun, আমি গরমের মধ্যে বেরোতে 25 ডিগ্রি উপরে চলে যাচ্ছি, টেম্পারেচার বাড়ছে, কোভিড তো হবে না, কোথায় ভাই, কোন জায়গায় হয়নি ভাই কোভিডটা? ইউনাইটেড আরব এমিরেটস অত হাই টেম্পারেচার, ইন্ডিয়া পুরো সামারে ছিল, আমাদের যেখানে ঠান্ডা সব জায়গাতে ছিল। ইট ইজ নট মেইনটেইনিং ইউর টেম্পারেচার, সো প্লিজ ডোন্ট বি সো মাচ হ্যাপি দ্যাট ইন্ডিয়াতে হচ্ছে না। অফকোর্স ইন্ডিয়াতে 6টা আলাদা ভ্যারাইটি আছে, মেবি মিউটেটেড ভার্সনটা ইন্ডিয়াতে যেটা এসছে হয়তো দ্যাট ইজ নট দ্যাট লিথল। হ্যাভিং সেড দ্যাট, আই উইল স্টিল টেল ইউ টু মেনটেন অল দ্য প্রিকশনস, প্লিজ মেনটেন দ্য প্রিকশনস as laid down in all the international bodies as well as in the Ministry of Health website. So thank you so much. I will be happy to take up any questions.
Thank you, sir, for your wonderful talk. It's really, really very interesting and informative for us and help to remove all the myths and misconceptions during this pandemic situation. Acha, I like to start our question answer session after this second talk. First question is been asked by Manali Bhattacharya.
Yes, tell me.
Okay. Nowadays, it is heard that a person who has already recovered from COVID-19 can still spread the virus up to 49 days. It is true. If yes, then how and why?
No, not at all. 49 days যে গল্পটা যেটা বলা হয়েছে, the thing which they have shown in the trials, there's actually the case reports, and you have to measure the IgG. If the patient is having IgG, please do not worry about that, whether the patient is having RT-PCR or not. If you go through all the good journal papers, good journal, I'm telling you, there are many predatory journals. Please do not go to the predatory journals. They are horrible. They publish anything like that. So any JAM or Lancet or Springer or any of the good journals which you have, go through the reinfection category, and then you will see that patients who had been reinfected, mostly they were reinfected because there were the other strains of there are already various mutated versions out there of this single virus. And the other one is that all of them, in fact from China itself, they have said very clearly, all of them did not show any infection transmission to their close contacts, as well as the HRCT scan was actually diminishing finally. So we are getting reports of reinfection in India. কিন্তু এখানে একটা গল্প আছে. Remember the discharge criteria of India. They do not recommend a generalized COVID-19 test to be negative before the patient is discharged. এটা কিন্তু ওয়েবসাইটে দেওয়া আছে. After 14 days, if you are out of the symptoms for 3 days, you can go back home and stay there for self-isolation for another 7 days. So 21 days you are there. You are not supposed to do a second COVID test. Of course, people are doing that, and many people are doing that. So what I would recommend, reinfection নাকি ওই পেশেন্টেরই RT-PCR তখন মানে আরো এক্সিস্টেন্স ছিল, পারসিস্টেন্স করছে, সেটাই হয়তো পারসিস্টেন্স পরে হয়তো নিয়েছে, হয়তো পেশেন্টের প্রথমবার অ্যাকচুয়ালি হয়েছিল প্রবলেম, মানে কোভিড-19 ছিল, অবভিয়াসলি ছিল, কারণ প্রথমবারটা পজিটিভ এসেছিল, সেকেন্ড টাইম ফিরে যাওয়ার পরে হয়তো নর্মাল কফ এন্ড কোল্ড হয়েছে, কিন্তু যেহেতু পেশেন্টের সেকেন্ড টাইম টেস্টটা হয়নি কোভিড-19 এবং পেশেন্টের পারসিস্টেন্স ছিল, মানে কোভিড-19 এর ভাইরাল ওই পিসিআরটা পারসিস্টেন্ট ছিল, কিন্তু অ্যাকচুয়ালি ব্যাপারে হয়েছে অন্য কোনো রোগ, মানে অন্য কোনো কারণে জ্বর, সেটা করতে গিয়ে পেশেন্ট আবার একটা RT-PCR করছে, আবার পজিটিভ এসে গেছে, পেশেন্ট ভাবছে রিইনফেকশন। সুতরাং there are many pros and cons before you say that there is a reinfection, and thankfully যে ডেটা আছে রিইনফেকশন থেকে আরেকজনের হবে না। So next question please.
Okay. Thank you, sir. Thank you, sir. Just a minute. Okay, next question is asked by Tirtharaj Paul. We have a huge confusion about the type of mask, especially by the news channel. So, sir, which type of masks will we provide as the maximum protection?
Maximum protection, of course, medical mask, nothing else. So there is no question about that. Okay, N95 is not recommended for anybody else other than healthcare workers. So you do not need to worry about that. A person who is sitting in front of you, if he is using a mask and I am using a mask, and both of them are using a cloth mask and properly placed, that's fine. That will reduce. So do not need to panic just like that. Next question.
Next question is asked by Preeti Jaiswal. After recovering from COVID, can RT-PCR test showing test positive? If yes, then, sorry?
Yes, just now I said.
Okay. If yes, then for how many days it will shows positive?
There is no data about that. Some of the data are there is no such data from India. There are data from, of course, from China and of course from all the world, where we can see up to stool PCR up to 57 days in children, up to 39 days in children in stool PCR. So does that mean that patient is ineffective? Does that mean the patient will spread the virus? thankfully no, because those are the patients who were RT-PCR positive, but I will go for viral culture, and viral cultures in those patients did not show any cytopathic changes which will indicate that the virus is replicating. So maybe that was a dead virus that was picked up. Next question.
Okay. Thank you, sir. Next question is asked by Bati Devnath. How long the antibody lasts after the first infection?
That's a very good question. In fact, right now I am measuring all of my patients. I am actually following them up. So I will give you a better idea after like 3 months, because I believe it will be after 39 months. It depends, it depends from various, on the various, like Mars-CoV titer raised for up to many months, okay? And SARS, it was again for like 6 months, 7 months. With this one, I guess it will be around 4 to 5 months or maybe 6 months. So the data is showing that the patients who had severe infection, of course, they will have the highest antibody, that is normal. And of course, they will be maintaining that titer for the longest time. As of date, what is the cutoff of this antibody? I do not know. If somebody can tell me, I'll be very happy. If some biologist, molecular biologist is there, so if you can tell me that what is the cutoff to say that this is the antibody titer which you must have for having protection. For me, I'm still following up my patients, and I guess it will be at least for around 3 months. Should be around for 3 months.
Thank you, sir. Thank you for all this, answering all this question. Thank you. Thank you for all your information, sir. I would like to again invite our first speaker to continue the question answer session.
Okay, go ahead. Go ahead.
Hello, sir.
Yeah, I'm on. Go ahead.
Okay. I'm asking the first question that is asked by Monali Bhattacharya. Depending on which this alpha beta types are distinguished.
Well, but sorry to say, I didn't. I don't have that answer because I didn't have to. I didn't. I didn't have this idea. Actually, there is a classifications already mentioned, but I cannot tell you. But obviously, it's there. The classification of the virus is obviously there. What basis they have classified alpha beta? Obviously, some sequence homology basically. But exactly answer I cannot tell you right now.
Thank you, sir, for your answer. The next question is asked by Rahul Mandal. What is R naught value?
R naught value. That is that one virus, the infect how many peoples at a time. So that is called the RG value. That is basically the virologist, the infection. That is a terms coined by the epidemiologist. So infected people can infect at a time how many persons? That's called the RG value. Average how many persons that they can infect.
Thank you, sir. The last question I'm taking, as the time is running out, that is asked by our ex-student Panchali Mukherjee. Is the R naught value is so low, then how this kind of infection occur? Like nowadays, it's about 55.
Hello. Yeah.
Can I repeat the question?
I didn't get your questions. Can you please?
Okay, sir. Yes, I am repeating it. If the R naught value is so low, then how this kind of infection occur? Like nowadays, it's about 55,000 in a day.
Well, that's really a good question. Think about that earlier epidemics. So earlier epidemics, whatever I discussed, so their R0 value is higher, but why it's not spread all over the world? Think about the informations of the infection. Once it is infection starts, the information was, they started taking the measures to, confine the infected people with some certain places. So that's why it was not allowed to spread. But think about the case of COVID-2. So whatever the data I have found, the first, the persons was infected in the Wuhan meat market as 2019 August. And 2019 to August, December actually, the information, spread all over the world that Wuhan has got some of this kind of virus infection. And WHO, I don't know, I'm not going to the controversies in the open here. WHO knew or they didn't know that, I don't know. But ultimately, that India started when? First December, that first people came from the Wuhan in the Kerala. He came. And then we start all this, the lockdown started March. But within this time period, many people from this China or the other part of the world already, they invaded in the India. And just take one example. So from August to December, so the statistics says almost 30 million, 30 lakhs people, visited USA within this 4 months. So think about, within this 4 months, 30 lakh, out of 30 lakhs, only 3 lakhs people carried the infection. They spread it almost 6 lakhs people in the USA. So, and that time you didn't take any measures. So obviously without the unconsciously, it has been spreaded. So that is the reason I think it has spreaded all over the world. So, but whose responsibility? I'm not going to then controversy, but this is the data suggests that RG value or infection ability is less compared to the previous epidemics.
Thank you, sir. Sir, again, a last question I'm asking.
Yeah.
It's asked by Abhishek Chatterjee, sorry. Is there any difficult antibody enhancement of entry of COVID and cytokine storm regarding immune therapy?
Well, I just showed actually Dr. Roy is there. He can, but whatever that I have told, still the FDA or whatever our ICMR didn't come out with any protocols. But without these things all over the world, the doctors have already, use some of the medicine, cytokines inhibitors, all of that, all the drugs are available, they used. And whatever the data came out, so with this data, whatever the positive data came out, with that information, some of the trials already, the going in all over the world. Just like I told you, some of the drugs already available, monoclonal antibody, already some of the monoclonal antibody treatment already available in the phase 2 or phase 1 or phase 2. Some of the drugs for the other purposes, that's also antiviral drugs are also, the phase 1 and phase 2 for the COVID. But the question of vaccines, I'm telling you, because still I'm telling you, I'm not that guy. I'm not that immunologist. I'm not that biologist. But again, whatever the knowledge we have from the laboratory, I can tell you that I told you that there are 80 different types of vaccines are started developing all over the world. Some of them are against the RNA, RNA against the antibody against the RNA. Some of the, and then most fruitful is the spike, the spike proteins. That's the, and then spike protein from the chimpanzees COVID-2. That is that the developing by the Oxford groups. And now the question is antibody, whatever the phase 3 trials again. But whether it will be fruitful or not, that we are still far from that reality. Because if you think about this, how many antibodies against the virus antibody successful? MMR, the smallpox or the polio, some of them. But there are lots of other viral infections. We could not make antibody. Influenza, we could not make antibody because every year the changes their antibody. We make, suppose the spike protein, you make an antibody of the spike protein, and next year you see this virus, it changes the the spike protein. Automatically the epitope antibody was generated against the epitope, that is not changed. That's not recognizing, that antibody is not recognizing that next year virus. So hopefully, still I think until unless it comes to the market, until unless it's the couple of years is with that vaccination, then we can come to the truth whether the antibody will be how much fruitful or not. So that is, as a I'm not a clinician, but I think the clinicians, Dr. Roy is there, he can light up more.
Well, Dr. Mukherjee, thank you so much for a beautiful elaboration. I actually saw your elaboration, that was fantastic.
Yeah.
So
thank you.
What I would rather tell you, I would rather try to share is that regarding the vaccine, of course, you're damn right that unless and until few years go, or at least few months there after the vaccine is there, we cannot say how much protection it will be giving. But from our general knowledge that we are getting from the influenza vaccine that we are prescribing regularly to our comorbid patients, we get a good amount of antibodies. So hopefully, again, I'm saying, different vaccines will generate different amount of antibodies depending on the type of vaccines we are using. Like if you're using a killed vaccine, it will generate, you might require 2 to 3 doses to achieve the same thing, rather than when you're using an adenovirus vector. So things will change in the type of the vaccine you're using, or the type of the vaccine, the transfer medium you are using. So all the things should be taken into consideration, and unless and until we have strong data, we cannot just presume. So this is something we should not be presumed. This vaccine, this is very important. This is the treatment for this all the viral, for most of the viral diseases. So another one, I think there was something about the cytokine inhibitors. There was some question about cytokine inhibitors, if I am not mistaken. Whether there were, can you tell me again there was any cytokine?
The question, cytokine inhibitors, cytokine.
Yes. Actually, my uncle was he was one of the patients who received that cytokine inhibitor. So we have been using that in many of the patients. Actimra, that is the tocilizumab, the interleukin-6 inhibitors. Recently, so this is actually the Roche company's product, and it failed its own company's product in phase 3 trial. But again, the trial was done in severely ill patients. So when you are looking into a study, please look into the inclusion criteria. This is a drug which should be given in a moderate patient, and finally going into severe, not a patient who is already severe. You will not get any benefit. Because a severe patient, we still do not know which drug will actually act. So we are in fact bombarded with all the drugs. Every day we are getting, this drug is acting, that drug is acting. In fact, famotidine has also claimed that it is acting. Dava Chyawanprash has also claimed that it is also acting. So I'm very much confused nowadays. I try to go by normally hydroxychloroquine, and generally with azithromycin if it's a mild to moderate. I do not know specifically what actually it will give, what result, because many of the patients who are not getting any drugs, they are just getting symptomatic relief. They are also getting cured. So what is the role of this kind of drug, or any drug in mild to moderate case, is still debatable. We need a randomized trial that should be double blinded. Then only we can say for sure that this drug works and this drug doesn't work. Like I'm just giving you one example. You're taking vitamin C and all those blah blah blah, all the famotidine and everything, and then you are wearing a PPE and you're seeing patients. Like I'm seeing a patient. Suppose I'm taking PPE. I'm taking all the universal precautions, and I'm taking vitamin C, and I'm not getting infected. Is effective? No, never. I have to open my PPE. I have to see the patients COVID-19 without any mask. Then if still I'm not getting infected, okay, vitamin C is acting. So please write up an article not by the conclusion, but read between the lines. Please see the whole text, not only the conclusion part. You will definitely get a wrong view. So this is very important. And regarding Actimra or interleukin-6, of course, I have seen clinical benefits, and not only me, many of the doctors have seen clinical benefits when it is given early at the onset of severe. So it's a moderate patient is coming to you, the saturation is decreasing like 90, 88, the patient is desaturating, and the patients have interleukin 6 very high, like 10 times, 10 times higher than your normal value. Go for the interleukin 6 inhibitor. At least you can say that you have tried my level best, and this is an experimental procedure. So of course you have to see the hepatic function as well as the renal function, and then you have to decide whether the patient should be given this or not. Thank you.
Thank you for such an informative and interesting talk and answering all these questions. Thank you. I'd like to thank both of our speakers for taking their time and effort for making such our webinar interesting and informative and sharing their great experience and thought with us. Thank you, sir. And
Thank
also
you.
I want to thanks on behalf of our Kalna College. Thank you. Now I'd like to invite our convener, Dr. Joydeep Acharya, to conclude today's session. Over to you, sir.
Thank you, Razina ma'am. I would like to thank Dr. Sudhir Shekhar Mukhopadhyay sir for appraising us about the structural and molecular formation as well as spread and preventing strategies of the virus through his great informative lecture. It will be very much helpful for us, particularly for the students of different branches of life science. Thanks to Dr. Shayok Roy for your energetic speech. Correct way of life we must adopt in order to combat this pandemic. Our honorable principal sir for giving his valuable time in our webinar busy schedule. Principal sir boosted us for the development of scientific temperament in our attitude, which is very much essential to fight against. By following his concept to nullify non-conjugate forces and fluxes, which are the harmful phenomenon, and nourishing the positive conjugate causes and effects, we are sure to reach our destination. Both the sessions of our first day program have been extremely informative, and we hope that it has eradicated irrational fears and formed a clear picture on the coronavirus, as well as it's taught us how to combat this pandemic. Before ending today's session, I'd like to announce that we are giving the link in your email ID as well as in the WhatsApp group to the participants. Please click yes to join for tomorrow. Now I would like to end today's session. Thank you all. Thank you again.
চন্দন, আমরা কজন লাস্ট অব্দি থেকে যাই?
চন্দন স্যার,
আচ্ছা বলো।
link.
Yes,
পাঠিয়ে দিয়েছি আমি।
it is already available in the group and also in the chat box.
It's okay. Anindita, can you hear me? Anindita. Nandita Ghosh. So I would now like to request all the participants to leave, because today's session has ended.